There is a world beneath the surface of the ocean where darkness is not merely the absence of light. It is a permanent condition, a state of being, a rule that has held for longer than any human civilization has existed. Below 3,000 feet, the sun does not reach. It cannot. The water above is too dense, too heavy, too absolute in its refusal to let photons pass. And yet, in that blackness, something glows, something pulses, something breathes light into the void, not from an external source, not from a star or a lamp or a fire, but from within its own living body. This is bioluminescence. And beneath 3,000 feet, it is not rare, it is not unusual, it is not the exception, it is the rule. Consider that statement for a moment. In the deepest, darkest, most inhospitable layer of the largest habitat on Earth, the dominant form of communication, defense, predation, and reproduction is light, organic light. Light manufactured by chemistry inside the cells of creatures that have never once experienced the warmth of the sun. Creatures that would die within minutes if they were brought to the surface. Creatures whose bodies are built for pressures that would crush the hull of a nuclear submarine. And yet they glow, blue, green, red, white, in flashes and waves and steady, haunting pulses that move through the water like slow-motion lightning. In bursts so brief, they last a fraction of a second, and in sustained emissions that endure for minutes without interruption, illuminating the emptiness around them in ways that human eyes were never designed to perceive. The deep ocean covers more than 60% of the planet's surface. It represents, by volume, the single largest living space on Earth. And within that space, according to scientific estimates compiled by researchers at some of the world's leading marine institutions, more than 76% of all observed marine organisms produce their own light. Not 76% of deep-sea creatures alone. 76% of all marine organisms that have been observed and catalogued. That means bioluminescence is not an exotic trick performed by a few strange species in the abyss. It is one of the most common biological traits in the entire ocean. More common than the ability to swim fast. More common than the ability to change color. More common than virtually any other single adaptation that marine science has documented. And most humans have never seen it. Most humans have never even heard of the creatures that produce it. The deep ocean is darker than any night sky, colder than any winter on the surface, and under more pressure than any environment humans have ever measured on land, and it is full of light. That is the paradox. That is the contradiction that has driven marine scientists to the edge of what they understand about biology, chemistry, and the very nature of life itself. How can a world without sunlight be filled with organisms that produce their own illumination? Why would the forces that shaped these creatures invest so heavily in a trait that seems, on the surface, unnecessary in total darkness? Who is the light for? What is it saying? And perhaps the most unsettling question of all, what else is down there, glowing in the silence, that we have not yet seen? We have mapped more of the surface of the moon than we have mapped of the ocean floor. We have sent more humans beyond the atmosphere of this planet than we have sent to the bottom of the deepest ocean trench. The technology required to survive at the very bottom of the sea is, in many respects, more complex and more demanding than the technology required to orbit the Earth. And the creatures that live in that environment, the ones that pulse and flash and shimmer in the eternal dark, are not waiting for our instruments to find them. They have been there, doing what they do, producing their light, hunting with it, hiding with it, communicating through it, for a span of time so long that it dwarfs any measure humans typically use. They were glowing before the first human being looked up at the stars. They will be glowing long after the last human satellite falls from orbit. By the end of tonight, you're going to understand why bioluminescence is not just a curiosity of the deep ocean. It is a window into one of the most profound mysteries of the natural world. A mystery that challenges what we think we know about darkness, about life, about the limits of biology, and about the vast, silent, glowing world that exists beneath our feet every single day. A world that is closer to us than the nearest city in many cases, and yet more alien, more strange, and more beautiful than anything that has ever been found beyond the atmosphere. Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It is a simple action, but it helps this channel reach more curious minds like yours. The ocean is not empty. It is not silent. It is not dead. Beneath the reach of sunlight, in a world of crushing pressure and freezing temperatures, something is shining. And what that light reveals is far stranger, far more beautiful, and far more unsettling than anything the surface world has ever produced. What that light conceals may be stranger still. There are organisms in the deep ocean right now producing light in colors that no other known creature can see. There are organisms flashing in patterns that do not correspond to any known function. There are organisms glowing continuously, burning through precious biochemical energy for reasons that no scientist has yet been able to explain. The deep ocean is not a place of simple survival. It is a place of elaborate, ongoing luminous performance, and the audience, if there is one, is invisible to us. Now, let's begin. To understand why the bioluminescent world beneath 3,000 feet is so extraordinary, it is first necessary to understand what 3,000 feet actually means. Not as an abstract number, not as a line on a chart, but as a physical reality, a place, a condition that the human body was never designed to endure, a depth at which the familiar laws of surface existence break down and are replaced by rules so harsh, so unforgiving, that even the most advanced engineering struggles to cope with them. The ocean is not a uniform body of water. It is layered, stratified, divided into zones that are as different from one another as a desert is from a rainforest. These zones are defined not by geography, not by temperature alone, but primarily by one factor: light. How much of it reaches a given depth determines nearly everything about what can survive there, how it behaves, and what it looks like. The ocean zones are, in the most fundamental sense, zones of illumination, and as you descend, the illumination does not merely decrease, it vanishes, it is extinguished, absorbed by the water molecule by molecule, wavelength by wavelength until nothing remains. The first zone is the epipelagic, sometimes called the sunlight zone. It extends from the surface down to approximately 200 meters, or about 650 feet. This is the ocean that most people know, the ocean of coral reefs and dolphins and surfers and cargo ships. Sunlight penetrates this layer with enough intensity to fuel photosynthesis, which means plants and phytoplankton can grow here. And because plants and phytoplankton form the base of the food chain, this zone is, by far, the most densely populated layer of the ocean. Nearly all commercial fishing takes place in the epipelagic. Nearly all human interaction with the sea occurs here. It is warm relative to the depths below. It is bright. It is familiar. When a child draws a picture of the ocean, they draw the epipelagic. When a filmmaker makes a documentary about marine life, the footage is almost always shot in the epipelagic. This is the ocean of public imagination, and it represents, in terms of the ocean's total volume, almost nothing. The epipelagic accounts for roughly 2 to 3% of the ocean's total living space. A thin skin of illuminated water stretched across a body of darkness so vast that it defies easy comprehension. Imagine a building 100 stories tall. The epipelagic is the top floor. Everything below it, 97 or 98 floors of water, is something else entirely. Something darker, something colder, something that operates by different rules. And the creatures that live in those lower floors have never seen the top. Below the epipelagic lies the mesopelagic zone, also known as the twilight zone. It extends from 200 meters down to roughly 1,000 meters, or about 3,300 feet. This is where sunlight begins to fail. Not all at once. Not like flipping a switch. But gradually, relentlessly, the light fades. At 200 meters, there is still enough illumination for a human eye to detect shapes and shadows. The water is a deep blue, almost indigo. By 300 meters, the blue has darkened to a purple so deep it is nearly black. By 500 meters, the light has dimmed to less than 1% of its surface intensity. A human eye at this depth would see nothing but the faintest suggestion of a difference between looking up and looking down. By 700 meters, even that suggestion is gone. And by 1,000 meters, the last photon of sunlight has been absorbed. The twilight zone is a place of transition, a slow, inexorable descent from the world of the sun into the world of the dark. And it is here, in this fading light, that bioluminescence begins to appear in earnest, not as a novelty, as a necessity. As the sun retreats, organisms must either follow the light upward, descend into darkness and learn to live without it, or create their own. The twilight zone is the boundary where that choice is made, and the overwhelming majority of organisms in the mesopelagic have chosen the third option. They make their own light. They carry it with them. They switch it on and off as needed. They use it for purposes that range from the practical to the mysterious. And they do so with a sophistication that rivals any technology humans have ever developed. The scale of life in the twilight zone has been one of the great revelations of modern oceanography. For decades, the mesopelagic was considered a biological desert, a zone of sparse, struggling life between the productive surface and the dead bottom. Scientists assumed that because sunlight faded in this zone, productivity would fade as well. They assumed that the biomass would decrease proportionally with the light. They assumed, in short, that the twilight zone was an empty transition, a corridor between the interesting upper ocean and the interesting bottom. This assumption was wrong, catastrophically wrong. A 2014 study published in the journal Nature Communications, based on acoustic surveys and net sampling conducted across the world's oceans, estimated that the biomass of mesopelagic fish alone is between 10 billion and 15 billion metric tons. That is roughly 10 times the previous estimate. 10 times more fish in the twilight light zone than anyone had thought existed there. And the reason the earlier estimates were so far off is instructive. Mesopelagic fish are small, typically between 2 and 10 centimeters in length. They are fragile, with delicate bodies and thin bones. They are extraordinarily good at avoiding nets, darting away from the pressure wave that precedes a trawl, and many of them undertake a daily vertical migration, rising hundreds of meters toward the surface at night to feed in richer waters, then descending back into the twilight zone at dawn to hide from predators. This migration is the largest movement of animal biomass on the planet. It happens every single day. It has been happening for longer than humans have been watching the ocean. And the organisms that make this journey are, almost universally, bioluminescent. Their bodies are studded with photophores, tiny light-producing organs that they use for counter-illumination, communication, species recognition, and predator evasion. The twilight zone is not empty. It is teeming with life, and that life is glowing. Below the mesopelagic is the bathypelagic zone, the midnight zone. It begins at 1,000 meters and extends down to 4,000 meters, roughly 13,000 feet. No sunlight reaches this layer. None. Not a single photon of solar origin penetrates to this depth. The darkness here is not the darkness of a closed room or a moonless night. It is not the darkness of a cave or a basement or the bottom of a mine shaft. Those darknesses are artificial. They are the result of blocking a light source that exists somewhere nearby. The darkness of the bathypelagic is natural. It is original. It is the darkness that existed before any light was made. It is absolute, total. The kind of darkness that has no reference point, no gradation, no subtle shift from dark to darker. It is simply black, uniformly, relentlessly, eternally black. If you were placed in the bathypelagic without a light source, you would not be able to see your own hand in front of your face. You would not be able to see the walls of the vessel around you. You would not be able to see anything at all. Your eyes would strain, your pupils would dilate to their maximum, and they would receive nothing. The only light that exists at this depth is the light produced by living organisms, and it is everywhere. To understand how different this darkness is from anything on the surface, consider the following comparison. On the darkest night, in the most remote location on Earth, with no moon and no artificial light for hundreds of miles, the human eye can still detect starlight. The faintest stars visible to the naked eye produce a measurable amount of illumination. Even on a heavily overcast night, atmospheric scattering of distant light sources produces a very faint glow that registers on sensitive instruments. In the bathypelagic zone, there is no such glow. There is no atmospheric scattering because there is no atmosphere. There is no starlight because there is no sky. There is only water pressing in from all sides, absorbing every photon that attempts to reach this depth from above. The darkness of the midnight zone is in the strictest physical sense more complete than any darkness that can be experienced on the surface of the Earth. It is the darkest natural environment that exists on this planet. and it covers an area larger than all the continents combined. The temperature in the bathypelagic drops to between 2 and 4 degrees Celsius, roughly 36 to 39 degrees Fahrenheit. This is not dramatically cold by terrestrial standards, but it is cold enough to slow metabolic processes significantly. Organisms in the midnight zone live in a state of perpetual near hypothermia by surface standards. Their movements are slow, their metabolisms are efficient, every calorie is precious, every chemical reaction is measured, and yet, despite the energetic cost, they produce light. They invest scarce biological resources in manufacturing photons. The fact that they do so, despite the cost, is perhaps the strongest evidence that bioluminescence is not a luxury in the deep ocean. It is a fundamental requirement. The pressure at this depth is immense, and its effects are not intuitive. On the surface, we live under one atmosphere of pressure. The weight of the entire column of air above us, from the ground to the edge of space, presses down on every surface with a force of roughly 14.7 pounds per square inch. We do not feel it because our bodies have evolved to operate at this pressure. It is our baseline, our normal. At 1,000 meters below the surface of the ocean, the pressure is approximately 100 atmospheres. That means every square inch of surface at that depth is bearing a weight of roughly 1,470 pounds. To put that in perspective, imagine stacking 14 full-size automobiles on top of a surface the size of a postage stamp. That is the kind of force pressing against every organism, every structure, every molecule of water at the top of the midnight zone. At 2,000 meters, the pressure has doubled to 200 atmospheres. At 3,000 meters, it is 300 atmospheres. The increase is linear and relentless. Every 10 meters of descent adds approximately one more atmosphere of pressure. There is no plateau, no relief, no depth at which the increase pauses or slows. It simply builds, accumulating force with every additional meter of water overhead. At 4,000 meters, the bottom of the bathypelagic, every square inch is enduring nearly 6,000 pounds of force. That is the weight of a large sport utility vehicle, concentrated on an area smaller than a human thumbnail. The effects of this pressure on human-made objects are dramatic and instructive. Styrofoam cups lowered to bathypelagic depths by oceanographers as a demonstration are crushed to a fraction of their original size, every air bubble in the foam compressed until the cup is no larger than a thimble. The demonstration is popular because it is visual and easy to understand, but it understates the reality. The styrofoam cup is made of a material that is mostly air. It compresses easily. A steel pressure vessel does not compress easily, but at sufficient depth even steel fails. The engineering challenge of building a vessel that can withstand bathypelagic pressures is one of the most demanding in all of materials science. The hull must be spherical, because a sphere distributes pressure evenly across its surface. It must be made of titanium or a specialized steel alloy, because ordinary structural steel lacks the strength-to-weight ratio needed for the task. And it must be machined to extraordinary tolerances, because even a small imperfection in the hull, a scratch, a void, a slightly uneven weld, becomes a stress concentration point that could initiate catastrophic failure under extreme pressure. The human body, unprotected, would not survive even the upper reaches of the midnight zone. The air in the lungs would compress to a fraction of its volume. The chest cavity would collapse. The sinuses, the eardrums, the soft tissues of the eyes would fail catastrophically. At the pressures found in the lower bathypelagic, death would not be a process. It would be an event. Instantaneous and total. The body would be compressed, deformed, reduced to something unrecognizable in less time than it takes to blink. Engineers who design pressure vessels for deep-sea submersibles describe the failure of an inadequate hull at depth as an implosion. The word is clinical. The reality is violent beyond description. The hull collapses inward at the speed of sound, converting the interior of the vessel into a compressed mass of metal and water in a fraction of a second. There is no warning. There is no gradual deformation. There is a vessel, and then there is not. And yet life thrives in the bathypelagic. Not just survives, thrives. The midnight zone is home to an estimated 1 million species that have never been catalogued. According to projections by the Census of Marine Life, a decade-long international research initiative that concluded in 2010. These are not primitive organisms clinging to the margins of existence. They are complex, sophisticated creatures with nervous systems, digestive tracts, reproductive strategies, sensory organs, and, in the vast majority of cases, the ability to produce their own light. They hunt, they evade, they mate, they communicate. They do all the things that surface organisms do, but they do it in conditions that would be instantly lethal to any creature from the world above. Below the bathypelagic lies the abyssopelagic zone, also called the abyssal zone. It stretches from 4,000 meters to 6,000 meters, roughly 13,000 to nearly 20,000 feet. The word abyss comes from the Greek abyssos, meaning bottomless, and while the abyssal zone does have a bottom, the name captures something essential about the psychology of this place. It feels bottomless. It feels like the end of the world. It feels like a place that should not contain life, that should not contain anything except water and rock and silence. Temperatures here are barely above freezing, hovering around 1 to 2 degrees Celsius. Pressure exceeds 400 atmospheres at the lower boundary. The water is still. Currents are weak and slow, measured in centimeters per second rather than the meters per second common at the surface. Sound travels differently here, bending and refracting through layers of water with different temperatures and salinities, creating a complex acoustic environment that organisms have learned to navigate. But light, visible light from the sun, is a memory that the abyss has never possessed, not in any meaningful quantity, not in any time frame that matters. Food is desperately scarce in the abyssal zone. In the epipelagic zone, a cubic meter of water might contain thousands of organisms and millions of individual cells. In the abyssal zone, a cubic meter might contain 1 or 2 organisms, or none. Life here is sparse, patient, and spectacularly adapted to conditions of extreme deprivation. Every calorie matters. Every movement is an investment that must yield a return or be abandoned. Organisms in the abyss do not chase food. They wait for food. They sit, motionless, sometimes for days or weeks, conserving energy, barely metabolizing until something edible drifts within reach, and then they act. Quickly, efficiently, decisively. The meal is consumed, and the waiting begins again. In the epipelagic zone, a cubic meter of water might contain thousands of organisms. In the abyssal zone, a cubic meter might contain 1 or 2, or none. Life here is sparse, patient, and spectacularly adapted. The primary food source in the abyss is what marine biologists call marine snow. It is a constant, slow, unending rain of dead organic matter drifting down from the sunlit world above. Fragments of dead phytoplankton, fecal pellets from zooplankton, shed mucus from jellyfish, the decaying remains of fish and crustaceans that lived and died thousands of meters overhead, discarded larval shells, the spent casings of diatoms, the broken filaments of colonial organisms that fell apart in midwater and began their long descent. These particles sink slowly, taking weeks or months to travel from the surface to the abyssal floor, tumbling through the water column at rates of 100 to 300 meters per day. By the time they arrive, much of their nutritional content has been consumed by organisms in the layers above, picked apart by filter feeders and scavengers at every intermediate depth. What reaches the abyss is the residue, the leftovers, the biological equivalent of crumbs falling from a table so high above that the floor cannot see it. And yet, this rain of death is the foundation of an entire ecosystem, a rain that never stops, that never pauses, that has been falling since the surface ocean first produced enough organic matter to generate a downward flux. The abyssal organisms that depend on marine snow have adapted to extreme scarcity. They have slow metabolisms, large mouths relative to their body size, and expandable stomachs that allow them to consume meals far larger than themselves when the rare opportunity presents itself. They cannot afford to be picky. In the abyss, you eat what falls, or you do not eat at all. And then, at the very bottom of the ocean's architecture, there is the hadal-pelagic zone, named after Hades, the Greek underworld, the realm of the dead, the place from which no traveller was expected to return. This is the zone of the ocean trenches, the deepest, most extreme, most inaccessible, and most mysterious environments on the planet's surface. The hadal-pelagic begins at 6,000 meters, and extends to the absolute floor of the ocean. In the Mariana Trench, that floor lies approximately 10,994 meters below sea level. That is nearly 11 kilometers straight down, nearly 7 miles of water, nearly 36,000 feet. To visualize this depth, consider that Mount Everest, the tallest mountain on Earth, stands at 8,849 meters above sea level. If you placed Mount Everest at the bottom of the Mariana Trench, its peak would still be submerged beneath more than 2,000 meters of water. More than a mile of ocean would still separate the summit of Everest from the surface. The depth of the ocean, at its most extreme, exceeds the height of the tallest mountain by a margin that is itself taller than many mountains on the surface. The pressure at the bottom of the Mariana Trench is approximately 1,086 atmospheres. That translates to roughly 16,000 pounds pressing against every square inch. If you took a standard steel shipping container, the kind you see stacked on cargo ships in ports around the world, and lowered it to the bottom of the Challenger Deep, the deepest known point in the Mariana Trench, the container would be crushed flat. Not dented, not warped, crushed flat. The steel walls would buckle inward like aluminum foil crumpled in a fist, and the process would take less than a second. The same force acts on every organism at that depth, and yet organisms exist there. They move, they feed, they reproduce, some of them produce light. The first humans to reach this depth were Jacques Piccard and Don Walsh, who descended to the floor of the Challenger Deep on January 23, 1960, aboard the bathyscaphe Trieste. The Trieste was not a submarine in the conventional sense. It was a deep-diving research vessel consisting of a large float filled with aviation gasoline for buoyancy, and a small, thick-walled pressure sphere suspended beneath it, barely large enough for 2 men. The descent took approximately 4 hours and 47 minutes. The 2 men spent only about 20 minutes at the bottom before beginning their ascent, partly because a crack had appeared in the outer window of the pressure sphere during the dive. They reported seeing a flat, featureless expanse of sediment, a lifeless-looking desert of pale, silty mud stretching in every direction. But even in that brief visit, even through the murky porthole of a vessel under unimaginable stress, they observed something unexpected. A flatfish, roughly 1 foot in length, resting on the bottom. Life, at the deepest point on Earth. Life, where no life was supposed to be able to exist. It would be more than 50 years before another human returned to that depth. In 2012, filmmaker James Cameron piloted the Deep Sea Challenger, a custom-built one-person submersible, to the floor of the Challenger Deep in a solo dive. Cameron spent approximately 2 hours and 36 minutes at the bottom. His vessel carried high-definition cameras, robotic sampling arms, and advanced lighting systems, designed to document an environment that had been visited by human eyes exactly once before, more than half a century earlier. And what those cameras captured was not a lifeless desert. It was a sparse but unmistakable ecosystem. Amphipods, tiny crustaceans no larger than a fingernail, scuttling across the sediment, microbial mats clinging to rocks. And, faintly, at the edges of the frame, barely detectable against the absolute blackness beyond the floodlights, the telltale shimmer of biological light. Something was glowing in the deepest trench on the planet. But these manned descents, while dramatic and historically significant, represent only a fraction of what scientists have learned about the deep ocean. The real work, the sustained, systematic, patient exploration of the deep, has been carried out by remotely operated vehicles, or ROVs, and autonomous underwater vehicles, or AUVs. These are robotic platforms, tethered to surface ships by long cables, or operating independently on pre-programmed missions, that can descend to depths no human can safely reach and remain there for hours or days at a time. They carry cameras, sampling arms, chemical sensors, and increasingly sophisticated instruments, designed to detect and measure bioluminescence without disturbing it. Institutions like the Monterey Bay Aquarium Research Institute, known as MBARI, founded in 1987 by David Packard, have spent decades sending these instrumented machines into the deep ocean. MBARI, located in Moss Landing, California, operates one of the most advanced fleets of deep-sea research vehicles in the world. Its ROVs, including the workhorse vehicles Ventana and Doc Ricketts, have logged more than 27,000 hours of deep-sea video footage over thousands of individual dives. They have captured images of creatures that no human eye had ever seen before. They have documented behaviors that no one had predicted. They have discovered entirely new species on a regular basis, sometimes multiple new species in a single dive. And one of the most consistent observations across all of that footage, across every dive, every depth, every geographic location, every season, every time of day and night, is the prevalence of bioluminescence. It is always there. It is always happening. The deep ocean is never dark in the way that a cave is dark or a closet is dark. It is illuminated constantly and continuously by the organisms that inhabit it. The illumination is faint by surface standards. A human eye, even fully dark adapted, might struggle to see some of the dimmer emissions. But the cumulative effect, the combined output of billions of individual organisms, each producing its own tiny share of light, is an environment that is not dark at all. It is a world lit from within. Edith Widder, a marine biologist and MacArthur Fellow who has spent more than 30 years studying deep-sea bioluminescence, has described the phenomenon as the most common form of communication on the planet. Not the most common form of communication in the ocean, on the planet. That statement, which she has made in published papers, in public lectures, and in her book Below the Edge of Darkness, published in 2021, is not hyperbole. It is a conclusion drawn from decades of direct observation and quantitative analysis. Her research, conducted through the Ocean Research and Conservation Association and through collaborations with institutions around the world, has demonstrated that bioluminescent organisms are found at every depth, in every ocean basin, and in almost every taxonomic group of marine life. Jellyfish glow, squid glow, fish glow, worms glow, crustaceans glow, single-celled dinoflagellates glow. Even some species of shark produce bioluminescent light from specialized cells in their skin. The phenomenon is so widespread, so deeply embedded in the biology of the ocean, that Widder has argued it should be considered not the exception, but the default state of marine life. In the ocean, producing light is normal. Not producing light is the oddity. This raises a question that sits at the heart of everything that follows. If the deep ocean is completely dark, and if the creatures living in it produce their own light, then who is watching? What is the light for? In the epipelagic zone, light serves obvious purposes. Sunlight fuels photosynthesis. It allows predators to spot their prey from a distance. It lets fish identify potential mates by color and pattern. But in a world without sunlight, where every photon, if self-generated, where every flash of illumination costs metabolic energy, the purposes of light become stranger, more complex, and far more difficult to understand. Why would an organism spend its limited energy producing light in a world where no external light exists? Who benefits? Who responds? Who is watching? The answer, as some scientists have gradually and painstakingly discovered, is that bioluminescence serves dozens of distinct and sometimes contradictory purposes. It is used for hunting, for defense, for mating, for communication, for camouflage, for warning, and for strategies so alien to our surface-dwelling experience that they barely have names in any human language. Some organisms use light to disappear. Others use it to attract attention. Some use it to signal potential mates across vast distances of dark water. Others use it to mark territory, to warn competitors, to confuse predators, or to illuminate the seafloor in search of food. The purposes are as diverse as the organisms that employ them, and new functions are still being discovered with almost every deep-sea research expedition. The deep ocean is not a world of darkness. It is a world of self-made light, governed by rules that humans are only beginning to comprehend, rules that are written in chemistry, enforced by pressure, and performed in a darkness so total that only the performers themselves can see the show. It is a world where light is currency, where photons are weapons, where a flash in the dark can mean the difference between eating and being eaten, between finding a mate and dying alone, between surviving and vanishing into the endless black without a trace. The foundation has been laid, the scale has been established. The ocean is deep beyond human intuition. It is dark beyond human experience. It is under pressures that exceed the limits of human engineering. It is cold, nutrient-poor, and separated from the surface by kilometers of water that act as a barrier more effective than any wall ever built. And in that impossible environment, life does not merely persist, it shines, it communicates, it performs. It has been doing so for longer than any surface ecosystem has existed in its current form, in an ongoing display that no audience has ever witnessed in its entirety. What comes next is the question of how. How does a living organism produce light from its own body? What chemistry drives this process? What structures house it? And why has this chemistry appeared independently, again and again, in organisms that share no common ancestor? The answer is far stranger than the darkness itself, and far more unsettling than the silence that surrounds it. The chemistry of bioluminescence begins with a single molecule. Its name is luciferin. The word comes from the Latin lucifer, meaning light-bearer, and despite the mythological associations of that name, the molecule itself is deceptively simple in concept if not in structure. Luciferin is a small organic compound, a substrate that produces visible light when it undergoes a chemical reaction with oxygen. The enzyme that catalyzes this reaction is called luciferase. Together, luciferin and luciferase form the biochemical engine that powers nearly all bioluminescence in the ocean. One molecule holds the potential for light, the other unlocks it. The reaction consumes oxygen, converts chemical energy into photon energy, and releases light with an efficiency that no human technology has been able to match. But here is the problem. There is not one type of luciferin. There are many. At least 11 distinct chemical forms of luciferin have been identified across the tree of life, and they are not all related to one another. Some are simple aldehydes, some are complex ring structures built around nitrogen and sulfur atoms, some require cofactors like calcium or magnesium to function properly. Some are synthesized internally by the organism that uses them. Others are obtained from food, passed up through the food chain, from organisms that manufacture them to organisms that merely consume and repurpose them. And the luciferases that activate these different luciferins are equally diverse, belonging to entirely different protein families, with no apparent common ancestor, no shared evolutionary origin, no structural similarity that would suggest they descended from a single ancestral enzyme. This means that bioluminescence did not arise once and spread across the ocean like a single invention shared among relatives. According to current scientific understanding, it arose independently, in separate lineages, on at least 40 different occasions. Some estimates place the number closer to 50 or even higher, 40 times, perhaps more, 40 separate instances in which unrelated organisms, living in different parts of the ocean, at different depths, under different pressures, facing different predators and different ecological challenges, independently develop the ability to produce light from within their own bodies using different chemicals and different enzymes. That is not a coincidence, that is a pattern. And the pattern suggests something profound about the deep ocean as an environment. Bioluminescence is not just advantageous there, it is in some fundamental sense inevitable. The conditions of the deep ocean are such that, given enough time, living organisms will arrive at the production of light, not because they are guided toward it, but because the environment rewards it so heavily that any lineage capable of producing light gains an advantage so significant that the trait persists and spreads. But why? What is it about the conditions beneath 3,000 feet that makes living light so essential that unrelated species keep arriving at the same solution independently, again and again, across different oceans and different geological periods? The answer lies in understanding what light does in a world without the sun. And the answers, as it turns out, are multiple, layered, and in some cases deeply disturbing. The first and most immediately understood function of bioluminescence is counter-illumination. This is a form of camouflage so ingenious that it borders on the paradoxical. In the mesopelagic zone, the twilight zone, there is still a faint amount of downwelling sunlight. It is dim, barely detectable to human eyes, but it is there, a faint, diffuse glow filtering down from the surface, growing weaker with every meter of descent, and for a predator looking upward from below, any organism swimming above it will appear as a dark silhouette against that faint glow. The shape of the body, the outline of the fins, the shadow of the belly. All of it is visible to a predator with sensitive enough eyes. And deep-sea predators have extraordinarily sensitive eyes. Their pupils are large, their retinas are packed with rod cells, the photoreceptors responsible for detecting low light levels. Some deep-sea fish have eyes that are more than 50 times more sensitive to light than human eyes. In the twilight zone, a silhouette that would be invisible to a human is a clear, obvious target to a predator adapted to the gloom. Counter-illumination solves this problem in an elegant way. The organism produces light on its underside, on the ventral surface of its body. That precisely matches the color and intensity of the downwelling sunlight above it. The result is that the silhouette disappears. The organism becomes invisible from below, not by hiding, not by fleeing, not by changing color in the conventional sense, but by generating its own light to erase its own shadow. It fills in the dark outline of its body with a glow that exactly replicates the light filtering down from the surface. The predator below looks up and sees an unbroken field of faint illumination. No shadow, no outline, no target. The hatchetfish, a small, flat-bodied fish common in the mesopelagic, is one of the most well-studied practitioners of counter-illumination. Its body is shaped like a blade, thin when viewed from the side, but broad when viewed from below, maximizing the surface area available for counter-illumination, while minimizing the silhouette visible from any other angle. Its ventral photophores, the light-producing organs embedded in its belly, can adjust their output in real time to match changing light conditions. If a cloud passes far overhead and the downwelling light dims slightly, the hatchetfish dims its photophores accordingly. If the sun breaks through and the light intensifies even marginally, the photophores brighten in response. The match is not approximate. It is precise to a degree that has astonished the biologists who have measured it. The fish is, in effect, wearing a suit of light that renders it invisible from below. A cloak of self-generated photons that perfectly cancels out the shadow its body would otherwise cast. And this is only the beginning. Counter-illumination operates in the twilight zone, where there is still some sunlight to match. Below the twilight zone, in the bathypelagic, there is no downwelling light at all. Counter-illumination is useless in total darkness because there is no ambient glow to mimic. There is no shadow to erase. There is only black. And so the purpose of bioluminescence shifts. It does not disappear. It does not become less important. If anything, it becomes more important. But its function changes from camouflage to something far more aggressive. It becomes a weapon, an active tool of predation, a means of locating, attracting, and consuming food in an environment where food is scarce, encounters with prey are rare, and every missed opportunity could mean starvation. And the most infamous example of this shift is the anglerfish. The anglerfish is, in many respects, the poster organism of the deep ocean. It appears in documentaries, in textbooks, in popular science articles, and in the nightmares of anyone who has ever seen a photograph of its face. It is ugly in a way that seems deliberate, almost theatrical, as if the darkness itself had designed a creature specifically to embody the concept of a nightmare-made flesh. Its body is round and bloated, often no larger than a grapefruit, but sometimes reaching the size of a basketball. Its skin is dark, often black or dark brown, absorbing what little light exists in its environment and returning nothing to the eye. Its mouth is disproportionately large relative to its body, sometimes exceeding the size of its own torso, and it is filled with long, translucent, needle-like teeth that curve inward at sharp angles, preventing anything that enters from ever escaping. The jaw is hinged to open to an extreme angle, far wider than any surface fish could manage, and the stomach is expandable, capable of stretching to accommodate prey larger than the anglerfish itself. In an environment where meals are rare, where weeks or months might pass between successful hunts, the ability to consume anything that comes close, regardless of its size relative to the predator, is not a luxury. It is a survival requirement. A single meal might sustain the anglerfish for weeks. A missed meal might be its last. But the anglerfish's most famous feature is not its mouth or its teeth or its expandable stomach. It is the elysium, a modified dorsal spine that extends from the top of its head like a fishing rod, arching forward over the skull and dangling a small, fleshy structure directly in front of its enormous mouth. At the tip of the Elysium is the esker, a bulbous lure filled with bioluminescent bacteria. The anglerfish does not produce its own light. This is a critical distinction. Unlike the hatchetfish, which generates its own photons through internal chemistry, the anglerfish outsources its light production to symbiotic bacteria. It farms light. It cultivates colonies of luminous bacteria within the specialized tissue of the esker, providing them with nutrients, with oxygen, with a stable environment protected from the surrounding water, and in return, the bacteria glow. The glow is soft, blue-green, steady or pulsing, depending on the species. And in the absolute darkness of the bathypelagic, where no other light exists except the self-generated flashes of other organisms, that glow is irresistible to smaller creatures. They approach it, drawn by what appears to be a tiny, helpless point of light in a vast, empty darkness, and they swim directly into the anglerfish's mouth. The trap is simple in concept. The execution is flawless. And the relationship between the anglerfish and its bacteria is one of the most well-known examples of symbiotic bioluminescence in the natural world. The bacteria gain a host, the anglerfish gains a lure, both benefit, and the prey, drawn by a light that promised food and delivered death, benefits not at all. But the anglerfish is not alone in using light as a weapon. The dragonfish, a family of deep-sea predators found throughout the world's oceans, has taken the weaponization of bioluminescence to a level that scientists once believed was impossible. The level of impossible. The level that requires, when you first read about it, a moment of pause to confirm that the description is accurate and not an error or an exaggeration. Most bioluminescence in the ocean is blue or green. There is a sound physical reason for this. Blue light has a short wavelength, between roughly 450 and 495 nanometers, and travels efficiently through seawater. Green light is similar, with wavelengths between roughly 495 and 570 nanometers. Most deep-sea organisms have eyes that are tuned to detect blue and green wavelengths, because those are the colors that bioluminescence typically produces and the colors that travel farthest through water. Red light, which has a longer wavelength between roughly 620 and 750 nanometers, is absorbed quickly by water. It does not penetrate to great depths. In the deep ocean, red light does not exist naturally, and because red light does not exist naturally in the deep sea, most deep-sea organisms cannot see it. They lack the photoreceptors necessary to detect red wavelengths. Their visual world is blue and black and shades of blue-green in between. Nothing else. Red is not a color that the deep ocean knows. The dragonfish exploits this blind spot with devastating effectiveness. Several species of dragonfish, particularly those in the genus Malacosteus and the genus Pachystomias, possess suborbital photophores, light-producing organs located just below their eyes that emit far-red light. Light with a wavelength of approximately 700 nanometers or longer. Light that is, to every other known organism in the deep sea, completely invisible. The dragonfish can see this light because it possesses a unique visual pigment derived from chlorophyll-based compounds, almost certainly obtained through its diet of copepods that consume chlorophyll-producing organisms. But its prey cannot see the light. Its predators cannot see the light. Nothing in the deep ocean, aside from the dragonfish itself and a handful of closely related species, can detect the wavelength it produces. The result is a predator with a built-in spotlight that only it can see, a biological infrared searchlight that illuminates its surroundings without alerting anything to its presence. The dragonfish swims through the dark, bathing everything around it in red light that is invisible to the organisms it hunts. It can see them clearly, their shapes and movements revealed by the reflected red glow, while they see nothing. They do not know they are being watched. They do not know they are being hunted. They continue their normal behaviors, oblivious to the predator that has them perfectly illuminated, perfectly targeted, perfectly in range. And then the dragonfish strikes. So this is not science fiction. This is documented biology. Researchers at Duke University, the Natural History Museum in London, and the Scripps Institution of Oceanography have studied dragonfish visual systems in detail. The far-red bioluminescence of Malacosteus was first described in scientific literature in 1984 by researchers who could barely believe what their instruments were telling them. It remains one of the most remarkable sensory adaptations ever discovered in any animal, in any environment, on land or in the sea. A predator that sees in a color that its prey cannot detect. A living stealth weapon, powered by chemistry, enabled by diet, and shaped by the relentless pressures of survival in total darkness. And it gets worse. The weaponization of light does not end with predation. It extends to defense. And the defensive strategies employed by deep-sea organisms are as varied, as creative, and as unsettling as anything found in the predator's toolkit. If predators have learned to use light to hunt, then prey have learned to use light to survive. The result is an arms race conducted entirely in photons, where every flash, every glow, every pulse of light is both a potential weapon and a potential shield. The vampire squid, Vampiroteuthis infernalis, whose Latin name literally translates to vampire squid from hell, is a small, deep-dwelling cephalopod found at depths between 600 and 1200 meters. It occupies a unique position in the classification of cephalopods, belonging to its own order, Vampiromorphidae, distinct from both the octopuses and the squids. Despite its terrifying name and its appearance, which features dark red skin, pale blue eyes, the size of a large marble, and webbed arms lined with fleshy spines called cirri, the vampire squid is not a predator in the traditional sense. It does not chase prey. It does not ambush prey. It feeds primarily on marine snow, using a pair of long retractable filaments, unique structures not found in any other cephalopod, to capture drifting organic particles from the water. It is, by deep-sea standards, a gentle creature, a scavenger, a collector of falling debris. But when threatened, the vampire squid deploys one of the most spectacular defensive displays in the animal kingdom. It can invert its web, turning its body inside out so that the dark outer surface is replaced by the spiny inner surface, presenting a larger, more intimidating silhouette to the predator. It can wave its arms in disorienting patterns, and most dramatically, it can produce bioluminescent mucus, a glowing cloud of sticky, luminous material that it ejects from the tips of its arms in a burst of cold blue-green light. The cloud hangs in the water, pulsing and swirling, its luminous particles slowly dispersing through the surrounding darkness. It persists for seconds, sometimes longer, a ghostly glowing mass with no solid body behind it. And while the predator investigates this phantom, the vampire squid goes completely dark. It shuts down every photophore, every luminous surface on its body. It retracts into its webbed arms, compressing itself into a tight dark ball that produces no light, no movement, no chemical signal. It becomes invisible in the deepest sense of the word, not just hidden, absent. The predator is left attacking a decoy, a phantom of light with no nutritional value, while the real organism drifts away in silence, consumed by the same darkness it has learned to weaponize. Other organisms use a similar principle in a more direct and violent way. Many species of deep-sea shrimp, particularly those in the families Oplophoridae and Sagestidae, have evolved a defensive response that is startling in its simplicity and devastating in its effectiveness. When seized by a predator, when teeth or tentacles or claws close around their body and escape by physical strength seems impossible, these shrimp release a burst of bioluminescent fluid directly into the face of their attacker. The fluid is produced by specialized glands near the mouth or along the body, and it is ejected in a cloud that envelops the predator's head, particularly its eyes. The sudden flash of light in an environment of total darkness is the biological equivalent of a flashbang grenade deployed at point-blank range. The predator's eyes, which have been painstakingly adapted for maximum sensitivity in near-zero light conditions, with pupils dilated to their widest and retinas loaded with photosensitive pigment, are suddenly flooded with an overwhelming burst of photons. The neural response is immediate and involuntary: pain, confusion, temporary blindness. The visual system, optimized for detecting the faintest possible glow, is overwhelmed by an input thousands of times brighter than anything it was calibrated to receive. And in the second that the predator flinches, in that single second of disorientation where its grip loosens and its attention fractures, the shrimp escapes. It jets backward, propelled by its tail, leaving the predator surrounded by a fading cloud of luminous fluid and a rapidly disappearing meal. Perhaps the most haunting and philosophically disturbing defensive use of bioluminescence is the so-called burglar alarm. This strategy is used by organisms that are too small, too slow, or too fragile to escape a predator through speed or strength or chemical defense alone. When attacked, these organisms do not try to hide. They do not try to blind their attacker. They do not flee. They do the opposite of what individual survival instinct would seem to demand. They produce the brightest, most conspicuous, most attention-grabbing burst of light they can manage, broadcasting their location to everything in the surrounding water. The purpose is not to scare the predator. The purpose is not to dazzle it or confuse it. The purpose is to attract an even larger predator. The flash of light is a beacon, a distress signal, a scream in the dark that says, in effect, something is hunting in this area, and here is exactly where it is. Come and see. For a large predator cruising through the deep ocean, a predator that has been searching for food in the vast, empty, dark expanse for hours or days, that beacon is an invitation, an opportunity. It means there is an active predator nearby, one that is currently occupied with a kill, distracted by its meal, and potentially vulnerable to ambush. The small organism, the one that triggered the alarm, may be consumed. Its individual fate may already be sealed, but its attacker now faces a threat of its own. Something larger has been called, something that the original predator would rather not meet. The burglar alarm turns the predator into prey. It sacrifices the individual to endanger the threat. It is, in the cold calculus of survival, an elegant and ruthless strategy. Edith Widder demonstrated this principle directly with one of the most celebrated and consequential experiments in deep-sea biology. In the early 2000s, she developed a device called the electronic jellyfish, or e-jelly, a small, battery-powered instrument designed to be attached to a deep-sea camera platform. The e-jelly consisted of a ring of LED lights programmed to mimic the bioluminescent display of the deep-sea jellyfish Atolla wivili. When threatened by a predator, Atolla produces a distinctive rotating pinwheel of blue light, a dramatic, spinning display that radiates outward in all directions like a slow-motion firework detonating in the darkness. The display is unmistakable. It is conspicuous. It is the most visible thing in the immediate environment for the several seconds it lasts. Widder hypothesized that this display was not a random panic response. She believed it was a burglar alarm, a deliberate, evolved signal designed not to scare the immediate attacker, but to attract something larger, something that would threaten the attacker and give the jellyfish a chance, however slim, to survive. Testing this hypothesis required going to the deep ocean and presenting the signal. Widder and her colleagues deployed the e-jelly on an unbaited camera platform near the Ogasawara Islands, south of mainland Japan. In a region of deep water known for its rich deep-sea fauna, the platform was lowered to a depth of approximately 900 meters. The e-jelly was activated, producing its spinning pinwheel of blue light in the blackness. And then the team waited, monitoring the camera feed from the surface. Within minutes, something approached. It was large. It moved with deliberate, powerful strokes, its arms trailing behind it in the current. It came directly toward the e-jelly, clearly attracted by the bioluminescent display. It was a giant squid, Architeuthis dux, one of the largest invertebrates on the planet, capable of reaching lengths of 13 meters or more. With eyes the size of dinner plates, the largest eyes in the animal kingdom, evolved specifically for detecting faint light in the deep ocean. The footage, captured in 2012 and subsequently broadcast around the world by the Discovery Channel and NHK, was a scientific sensation. It was among the first times a living giant squid had been filmed in its natural habitat, in the deep ocean, behaving naturally. Previous footage, captured in 2004 by Japanese researchers Tsunemi Kubadera and Kyoichi Mori, had shown a giant squid attacking a baited line. But Widder's footage showed something different. It showed a giant squid responding to light, responding to a signal, coming when the bioluminescent alarm was sounded. The implications were extraordinary and deeply unsettling for anyone who had assumed the deep ocean was a simple, quiet place. Giant squid, organisms that had evaded human observation for centuries, despite being among the largest invertebrates on Earth, despite washing up dead on beaches with enough regularity to fuel centuries of sea monster mythology, had been responding to the bioluminescent language of the deep ocean all along. They were listening to the light, they were interpreting it, they were reading the signals of other organisms and acting on that information. The deep ocean was not a void populated by isolated solitary creatures bumbling through the darkness. It was a network of luminous communication, a web of light-based signals connecting organisms across species and across trophic levels, and the giant squid was part of it, an active participant, a reader of the light who could be called out of the darkness by the right signal at the right time. And no one had known. For all of human history, the giant squid had been a legend, a mystery, a creature glimpsed only in death. And the key to finding it alive had been, all along, speaking its language, speaking in light. But here is the problem. For every function of bioluminescence that scientists have identified and explained, for every counter-illumination strategy, every lure, every flashbang defense, every burglar alarm, there remain hundreds of observed bioluminescent displays that have no clear explanation. Deep-sea ROV footage, accumulated over decades of expeditions by MBARI, NOAA, the Schmidt Ocean Institute, and dozens of other research institutions, is filled with luminous events that do not fit neatly into any known category. Flashes that seem too brief to serve as lures, glows that seem too dim to function as alarms, patterns that repeat at regular intervals with no apparent trigger and no apparent audience. Organisms that produce light continuously, without stopping, hour after hour, burning through biochemical energy at measurable metabolic cost, for reasons that no one can yet explain. The comb jellies, the siphonophores, the larvaceans, and the salps, and the countless unnamed, gelatinous organisms that drift through the midwater column all produce light in ways that defy easy categorization. The comb jellies, or catennophores, are a particularly confusing and beautiful example. These fragile, gelatinous organisms are found throughout the water column, from the surface to the deepest trenches, and many species produce stunning displays of bioluminescence when disturbed. Their bodies are transparent, sometimes nearly invisible in the water, and they move by beating rows of cilia, hair-like structures arranged in 8 longitudinal bands called comb rows. These cilia beat in coordinated waves, propelling the organisms slowly through the water. But comb jellies are famous for another optical effect that is often confused with bioluminescence: the shimmering rainbow-like bands of light that ripple along their comb rows as the cilia beat. These rainbow bands are not bioluminescence. They are not produced by chemistry. They are caused by the diffraction of light, the same physical process that creates rainbows in the sky after a rainstorm, or the iridescent sheen on the surface of a soap bubble. Each cilium acts as a tiny prism, splitting light into its component wavelengths, and as the cilia beat in sequence, the rainbow effect travels along the comb row in a mesmerizing wave. In the sunlit upper ocean, this diffraction is powered by ambient light. The cilia scatter sunlight into moving bands of color. It is beautiful, but it is physically straightforward. In the deep ocean, however, there is no ambient light to scatter. The only light available is whatever the comb jelly itself produces through bioluminescence. And here is where the mystery deepens. The organism creates its own light through a chemical reaction, and then it diffracts that light through its own body, splitting its own self-generated photons into a rainbow that moves along its comb rows in rippling waves of color. The organism creates its own rainbow. It generates the light, and then fractures it into a spectrum that only itself, and perhaps a handful of nearby organisms, can see. No one knows why it does this. No one has proposed a convincing explanation for why a deep-sea comb jelly would invest energy in producing light and then invest further biological architecture in splitting that light into a moving, multicolored display with no known audience and no demonstrated function. And the comb jellies are far from the only source of bioluminescent mystery. There are organisms in the deep ocean that produce light in wavelengths that no known deep-sea eye can detect. Not red light, like the dragonfish uses, wavelengths even further outside the range of any documented visual system. The purpose of light that cannot be seen by any organism in the environment is, to put it plainly, a mystery that challenges the basic assumptions of how bioluminescence is supposed to work. It is as if a human were to build a radio transmitter that broadcasts on a frequency no receiver can pick up. The energy is expended, the signal is sent, but no one is listening, or at least, no one that scientists have identified. Perhaps there are organisms with visual capabilities that have not yet been discovered. Perhaps the light serves a function that is not visual at all. Perhaps it is a byproduct of another chemical process, a waste emission with no communicative purpose. But the consistency with which it appears across multiple species in multiple regions of the ocean suggests otherwise. Waste products are random. This is not random. There are deep-sea worms in the family Tomopteridae that produce yellow bioluminescence. Yellow is exceedingly rare in the deep ocean. Most bioluminescence is blue or green because those wavelengths travel farthest through water. Yellow light is absorbed more quickly and travels shorter distances. And yet these worms produce it. They secrete yellow luminous particles from their parapodia, the paddle-like appendages along their sides, creating a trail of golden light as they swim. The particles hang in the water for several seconds before fading. The function of this display has never been satisfactorily explained. It may serve as a defensive distraction, like the vampire squid's luminous mucus. It may be a form of territorial marking. It may be a mating signal, or it may serve a purpose that scientists have not yet imagined. The yellow worms of the deep are one of dozens of examples of bioluminescent phenomena that have been observed, documented, filmed, and analyzed without yielding a clear answer. There are also organisms that produce bioluminescence in response to mechanical stimulation, but only in certain parts of their body, creating precise, localized patterns of light that seem designed to convey specific information. Some sea pens, colonial organisms related to corals that anchor themselves in the sediment of the abyssal floor, produce waves of bioluminescence that travel along their length when touched. The wave starts at the point of contact and propagates outward in both directions, reaching the tips of the colony and then reflecting back. The speed of the wave, its intensity, and the number of times it reflects can vary. Some researchers have suggested that these variations encode information about the nature and severity of the threat. A gentle touch produces a slow, dim wave. A vigorous disturbance produces a fast, bright cascade. The sea pen may be communicating through its bioluminescent wave, not just that it has been touched, but how it has been touched and by what. The scale of what remains unknown is staggering. Marine biologists estimate that more than 90% of the organisms in the deep ocean have never been described by science, never been named, never been classified, never been examined under a microscope or analyzed in a laboratory. They exist as shadows on sonar screens, as fleeting flashes in ROV camera feeds, as smears of bioluminescent light in the darkness that vanish before the camera can focus. Some of these organisms are so fragile, so delicate, that the water turbulence created by an approaching ROV destroys them before they can be photographed. They are creatures made of water and protein and light, barely distinguishable from the medium in which they live, and they dissolve at the first touch of anything solid. The only evidence of their existence, in many cases, is the flash of light they produce as they are disturbed and destroyed. A single flash, recorded by a camera for a fraction of a second, and then gone. That flash is, for many deep-sea organisms, the only record that they ever existed at all. And with each new expedition, with each new ROV dive, with each new camera deployment in a previously unstudied region or at a previously unexplored depth, the catalogue of unexplained bioluminescent behaviors grows longer, not shorter. Longer. Every expedition adds more observations that cannot be explained by existing models. Every new depth record reveals organisms that have never been seen before. Every technological improvement in camera sensitivity, in spectral range, in low light detection capability, reveals layers of bioluminescent activity that were previously invisible even to the instruments themselves. Scientists are not converging on a complete understanding of deep-sea bioluminescence. They are diverging from one. The more they look, the more they see. And the more they see, the less certain they become about how much remains unseen. The core problem is not that scientists do not understand bioluminescence. They do, at least in part. They understand the chemistry at a molecular level. They understand many of the ecological functions in specific species. They have catalogued hundreds of bioluminescent organisms and dozens of distinct strategies. They can explain counter-illumination. They can explain the anglerfish's lure. They can explain the burglar alarm. These are real achievements built on decades of difficult, expensive, often dangerous fieldwork. The core problem is that the more they learn, the more they realize how much they do not know. Every answer generates new questions. Every discovery reveals a deeper layer of complexity. Every expedition returns with more footage of unexplained phenomena than footage of phenomena that fit existing categories. The deep ocean is not surrendering its secrets willingly. It is revealing them one photon at a time, in flashes too brief to photograph and patterns too complex to decode. And behind every revelation, in the darkness just beyond the camera's range, always just beyond the camera's range, there is more. More light, more organisms, more behaviors, more questions. Always more questions. There is a concept in the philosophy of science called the expanding horizon of ignorance. It holds that as the boundary of knowledge grows outward, the boundary of unknown territory grows even faster, because each new discovery opens up new avenues of inquiry that did not exist before. In no field of science is this concept more vividly illustrated than in the study of deep-sea bioluminescence. The horizon is expanding, the ignorance is growing, and the ocean, vast and dark and patient, continues to glow. Consider the sheer arithmetic of the problem. The deep ocean, defined as water deeper than 1,000 meters, contains approximately 1.3 billion cubic kilometers of water. Humanity's total cumulative ROV and submersible observation of the deep ocean, across all nations, all institutions, all decades of exploration combined, has directly observed a volume equivalent to a few swimming pools relative to the size of the Pacific Ocean. The percentage of the deep ocean floor that has been surveyed at high resolution is in the low single digits. The percentage of the deep water column that has been systematically monitored for bioluminescent activity is effectively zero. Every statement that scientists make about the prevalence, function, or diversity of bioluminescence in the deep ocean is an extrapolation from a sample so vanishingly small that in any other scientific discipline it would be considered statistically meaningless. And yet the extrapolations themselves are astonishing. If 76% of the organisms in the small fraction of the ocean that humans have observed are bioluminescent, and if the unobserved regions are even remotely similar, then the total number of bioluminescent organisms on this planet is a figure so large that the human mind struggles to hold it. Trillions upon trillions of individual organisms, all producing light, all signaling, all communicating, all participating in a web of luminous exchange that spans every ocean basin on Earth, from the Arctic to the Antarctic, from the shallows of continental shelves to the deepest trenches, every night, every hour, every second. Right now, as these words are spoken, the deep ocean is glowing. It has been glowing since before there were eyes to see it, and it will continue to glow long after every human question about it has been asked and forgotten. But here's what makes this truly terrifying. The bioluminescent world beneath 3,000 feet is not a static display. It is not a gallery of frozen specimens waiting to be catalogued. It is not a museum exhibit that can be documented and closed. It is a living conversation, a continuous, ongoing, dynamic exchange of information between organisms that have been communicating in light for longer than any human language has existed, longer than any human civilization has stood, longer perhaps than the current arrangement of uncontinents has persisted. And we have only just begun to listen. We have pressed our ears to the wall of the deep ocean for a few brief decades, heard fragments of a symphony that has been playing for an unimaginable span of time, and we are still trying to identify the instruments. We cannot name the players. We cannot read the score. We do not know how many movements the symphony contains, or whether what we are hearing is the overture, the crescendo, or the final diminishing notes of a performance that began before our species learned to walk upright. All we know is that the music is real, the light is real, the conversation is happening, has been happening, and will continue to happen whether we understand it or not. The deep ocean does not need our comprehension, it only needs its darkness, its chemistry, and its time. The history of human attempts to observe bioluminescence in the deep ocean is a history of ingenuity, frustration, and repeated humbling. It is a story of brilliant minds devising extraordinary machines to travel to extraordinary places, and then discovering, upon arrival, that the very act of observation was destroying the thing they had come to see. Every light source brought to the deep ocean overwhelmed the delicate bioluminescent signals. Every engine vibrated the water and scattered the organisms. Every camera was either too slow, too insensitive, or too narrowly focused to capture what was happening in the darkness beyond its frame. The history of deep-sea bioluminescence research is, in many ways, a history of learning how to stop interfering with the phenomenon long enough to see it. The first human to witness bioluminescence in the deep ocean with his own eyes was almost certainly William Beebe. In 1930, Beebe and his engineer Otis Barton descended into the waters off Bermuda inside a steel sphere called the bathysphere. The vessel was crude by modern standards. It was essentially a hollow steel ball, 4 feet 9 inches in interior diameter, with walls 1.5 inches thick, equipped with 2 small quartz windows 3 inches across, and a telephone line connected to the surface. There was no propulsion system. The bathysphere hung from a steel cable attached to a winch on a barge above. It could go down, it could come up. It could not maneuver, steer, or reposition itself in any direction. Beebe and Barton were passengers, not pilots. They went where the cable took them, saw what the current brought to their windows, and hoped that the cable would not snap. Between 1930 and 1934, Beebe made a series of descents in the bathysphere, eventually reaching a record depth of 3,028 feet, deeper than any human being had ever gone before. What he saw through those tiny quartz windows would occupy him for years afterward. He described luminous organisms of every conceivable shape and size, creatures that he could not identify, and in many cases could not even compare to anything in existing zoological literature. He saw what he called a large, dim, pale bluish light that moved slowly past the window at a depth of around 2,000 feet. He described chains of luminous dots, spiraling patterns, sudden brilliant flashes that appeared and vanished in an instant. He saw organisms that seemed to produce light in response to the bathysphere's searchlight, and organisms that seemed to flee from it. He described the experience as overwhelming. There was too much to see, too much happening at once, and the windows were too small and the dives too brief to make systematic observations. Beebe's descriptions were vivid, detailed, and frequently dismissed by the scientific establishment. He was a popularizer as much as a scientist. He wrote for general audiences. He appeared on radio programs. He was, in the eyes of many of his academic contemporaries, a showman first and a researcher second. Some of his descriptions of deep-sea organisms, particularly the larger ones, were met with skepticism that bordered on open ridicule. He described a fish more than 6 feet long with luminous markings that did not match any known species. He described what he called a pallid sailfin, a large pale fish with prominent dorsal and anal fins that glowed with a soft luminescence. No such organism has ever been captured or photographed. To this day, some of Beebe's observations remain unconfirmed. They exist in a peculiar scientific limbo, neither verified nor definitively debunked, a reminder that the deep ocean is capable of producing organisms that a single observer can describe, but an entire scientific community cannot confirm. What Beebe understood, however, and what many of his critics did not, was that his own presence was distorting his observations. The bathysphere's searchlight, necessary for visibility, was a catastrophically bright intrusion into a world of total darkness. It was the equivalent of parking a helicopter with its spotlight aimed downward in the middle of a forest at night, and then attempting to observe the natural behavior of nocturnal animals. Everything within the searchlight's beam was illuminated, exposed, and almost certainly behaving abnormally. Everything outside the beam was invisible. Beebe was aware of this problem. He wrote about it. He understood that the bioluminescent displays he observed through his windows might be distress signals, defensive reactions, or behaviors triggered entirely by the bathysphere's presence, rather than natural behaviors occurring in the undisturbed darkness. But he had no solution. There was no way, with the technology of the 1930s, to observe the deep ocean without bringing light into it, and Beebe saw things that haunted him. On one dive, descending through the bathypelagic zone, he described a coordinated display of lights at a distance from the bathysphere that he could not precisely estimate. Dozens, perhaps hundreds, of individual luminous points arranged in a pattern that seemed deliberate. They moved together. They pulsed together. They maintained their formation as they drifted past the window. Beebe could not identify the organisms producing them. He did not know whether he was looking at a single large organism, a school of smaller organisms moving in coordination, or something else entirely. He recorded the observation. He noted his uncertainty, and he moved on to the next wonder, because the deep ocean gave him no time to dwell. Every second of descent brought something new, something unseen, something that no human being had ever witnessed before. He was the first to see any of it, and he was acutely, sometimes painfully, aware that he could not see enough. The limitations of Beebe's observations were not merely technological, they were fundamental. A human eye, even a trained and experienced one, can only process so much visual information per second. The deep ocean, viewed through a tiny quartz window during a descent that lasted less than an hour, was delivering more information than any human brain could absorb. Beebe described the experience as a kind of sensory overload in which he was constantly forced to choose between watching one extraordinary phenomenon and turning to observe another that was already vanishing beyond the window's edge. He could not photograph most of what he saw. The cameras of his era were too slow, their film too insensitive to light, their operation too cumbersome within the cramped interior of the bathysphere. Many of his most remarkable observations exist only in his written accounts and sketches, mediated by the imperfections of human memory and the inherent subjectivity of verbal description. This is one of the quiet tragedies of early deep-sea exploration. The first eyes to see the bioluminescent world of the deep ocean were human eyes, beautiful and sensitive, but fundamentally inadequate for the task. They could detect light. They could perceive color within limits. They could track motion, but they could not record. They could not replay. They could not zoom, enhance, or stabilize. They could only watch, remember, and describe. And the gap between what was seen and what could be conveyed in language was immense. This problem would persist for decades. When the Trieste reached the bottom of the Mariana Trench in 1960, Jacques Piccard and Don Walsh peered through their viewport into a world illuminated by their vessel's floodlights. They saw the bottom. They saw sediment. They reported seeing what appeared to be a flatfish on the trench floor, though this observation was later disputed. But they could not observe bioluminescence because their own lights drowned out any natural luminescence in the environment. They were searching for stars while carrying a sun. The advent of remotely operated vehicles in the 1970s and 80s brought new capabilities but also new problems. ROVs were smaller than manned submersibles, more maneuverable, and could stay submerged for longer periods. They could carry high-quality cameras and were operated from the relative comfort and safety of a surface vessel. But they were also loud. Their thrusters churned the water. Their tether cables dragged through the current. Their lights, essential for the cameras, were powerful halogen or later LED arrays designed to illuminate the deep-sea environment for visual observation, and their presence triggered exactly the same problems that Beebe had identified half a century earlier. Organisms fled. Organisms displayed defensive bioluminescence. Organisms that normally moved slowly and deliberately were startled into rapid escape responses. The ROV was observing a disturbed ecosystem, not a natural one. The scale of this disturbance was not fully appreciated for many years. Early ROV operators understandably focused on the technical challenges of deploying and controlling a complex machine in an extreme environment, often treated the behavioral responses of organisms as curiosities rather than as artifacts of observation. When a deep-sea organism flashed in the presence of an ROV, it was recorded as bioluminescence. The question of whether the organism would have produced that same flash in the absence of the ROV was rarely asked. When a camera recorded an apparently empty patch of water, and then, seconds later, an organism appeared at the edge of the frame, the organism was catalogued as present. The possibility that the organism had been attracted by the lights, or that it had just entered the camera's view while fleeing from the ROV's noise, was not systematically considered. For decades, the scientific picture of deep-sea bioluminescence was built on observations contaminated by the observer effect, a problem familiar from quantum mechanics but rarely discussed in marine biology. The deep ocean that ROVs showed us was not the deep ocean as it exists. It was the deep ocean reacting to our presence. The two are not the same. It was Edith Widder who first developed a systematic approach to solving this problem. In the 1990s, Widder began developing cameras and observation platforms designed specifically to minimize the observer's impact on the deep-sea environment. Her work was driven by a simple but radical premise. If you want to see what happens in the dark, you must become part of the dark. You must stop bringing light. You must stop making noise. You must stop moving through the water. You must make yourself invisible, indistinguishable from the darkness itself, and wait for the deep ocean to reveal what it does when no one is watching. Her first major innovation was the use of far-red illumination. Most deep-sea organisms cannot see red light. Their visual systems are adapted to detect blue and green wavelengths, the colors of bioluminescence and the last remnants of sunlight that penetrate to the mesopelagic zone. By illuminating the environment with far-red light, Widder could observe organisms without triggering the defensive or avoidance behaviors caused by white light or blue light. She was, in effect, using a wavelength of light that was invisible to the organisms she was watching. She could see them. They could not see her light. For the first time, it was possible to observe deep-sea organisms behaving naturally in the presence of an artificial light source. Widder's second major innovation was the Eye in the Sea, or EITS, a camera platform designed to be deployed on the ocean floor and left there, unattended, for extended periods. The EITS used far-red illumination and was equipped with an intensified camera system capable of detecting extremely faint bioluminescent emissions. It had no thrusters, no tether, no moving parts that could vibrate the water. It sat on the bottom like a rock, silent and dark, and waited. When triggered by a timer or by the detection of motion, it would begin recording. The goal was to capture the deep ocean as it actually was when humans were not present, to catch the inhabitants of the abyss going about their ordinary lives, communicating in light, hunting, feeding, mating, dying, all without the interference of an observer. The results were remarkable. The EITS captured footage of organisms and behaviors that had never been recorded before. Organisms approached the camera platform, investigated it, and in some cases displayed bioluminescent patterns that had no parallel in the existing scientific literature. Unknown species appeared. Known species behaved in unfamiliar ways. The footage revealed a deep ocean that was far more dynamic, far more active, and far more visually complex than the picture painted by decades of ROV and submersible observations. The simple act of removing the observer, of making the camera quiet and dark and patient, had opened a window into a world that had been there all along, but had been hidden by the very methods used to search for it. The difference between what the ETS recorded and what ROV cameras typically captured was not subtle, it was categorical. The ROV footage showed a disturbed environment, organisms reacting, fleeing, defending. The EITS footage showed a resting environment, organisms behaving as they behaved when no one was watching, feeding, communicating, interacting with one another in ways that suggested established relationships, familiar territories, and ongoing exchanges of information through light. The EETS footage was, in a very real sense, the first honest look that any human had ever had at the undisturbed bioluminescent world of the deep ocean. It was not a clearer view of the same picture. It was a different picture entirely. And it raised the unsettling possibility that everything scientists thought they knew about deep-sea bioluminescent behavior, every observation made from a lit, noisy, vibrating platform, every flash recorded in the beam of an ROV's searchlight, every behavior catalogued during the brief, intrusive visits of manned submersibles, might have been an artifact, not the real thing. a reflection of the deep ocean's response to human presence, rather than a window into the deep ocean's actual life. Widder also developed what she called the splat camera, short for submersible profile lensing array for thin layers. This was designed to profile the water column, recording bioluminescent activity at different depths as it was lowered through the water on a cable. The splat camera used a large, sensitive lens array to capture the faintest possible bioluminescent emissions across a broad field of view. It operated in complete darkness, with no artificial illumination, recording only the light produced by the organisms themselves. The data it returned were revelatory. The water column was not uniformly luminous. Bioluminescent activity was concentrated in layers, in thin horizontal bands, where organisms congregated at specific depths, presumably drawn by food resources, temperature gradients, or other environmental factors. The deep ocean was stratified not just by temperature, salinity, and pressure, but by light. There were layers of luminous intensity and layers of relative darkness, and the boundaries between them shifted over the course of the day and night cycle as organisms migrated vertically through the water column. This vertical migration, the daily movement of organisms between deep water and shallow water, is itself one of the most extraordinary biological phenomena on Earth. Every day, as the sun sets, billions upon billions of organisms begin rising from the deep ocean toward the surface to feed in the plankton-rich waters of the upper ocean. At dawn, they descended again, retreating to the darkness of the mesopelagic and bathypelagic zones. This migration, known as the diel vertical migration, is the largest migration of living organisms on the planet. It happens every day. It involves biomass measured in gigatons, and much of it is conducted by bioluminescent organisms that produce light as they move, creating a slow, massive, luminous pulse that rises and falls through the water column every 24 hours. Imagine a wave of light, hundreds of meters thick and spanning entire ocean basins, rising from the darkness every evening and sinking every morning. That is the diel vertical migration, seen through the lens of bioluminescence. The numbers are striking. Current estimates suggest that the total biomass involved in the diel vertical migration exceeds 10 billion metric tons, making it the single largest coordinated movement of living matter on the planet by several orders of magnitude. The diel vertical migration has been documented for over a century, first as a mysterious deep scattering layer on naval sonar screens during the Second World War. Sonar operators detected what appeared to be a false bottom, a reflective layer in the water column that rose toward the surface at dusk and descended at dawn. For a time, the layer was considered a sonar artifact, a phantom feature generated by the equipment itself. But the phantom was consistent. It appeared on every sonar screen, in every ocean, at every latitude. It moved, it breathed, it responded to the cycle of day and night with a regularity that no equipment malfunction could explain. Eventually, researchers determined that the deep scattering layer was biological. It was composed of countless organisms, fish, squid, crustaceans, jellies, all migrating vertically through the water column in synchrony with the sun. Many of these organisms are bioluminescent. As they rise, they carry their light with them. As they descend, they take it back. The deep scattering layer is not just a sonar signature. It is a luminous migration, a nightly river of living light that flows vertically through every ocean on Earth. And despite decades of study, the exact role that bioluminescence plays in coordinating, facilitating, or complicating this migration remains poorly understood. Does light production help organisms navigate during the migration? Does it help them find one another, maintain school cohesion, avoid predators during the vulnerable hours of transit through illuminated waters? Or does it compromise their safety, making them visible to predators from above and below during the most dangerous hours of their daily cycle? The answers are not clear, and the difficulty of observing bioluminescence during a migration that spans hundreds of meters of water column across thousands of square kilometers of ocean surface makes experimental investigation extraordinarily challenging. But the technology of observation has improved in ways that extend far beyond cameras. The development of autonomous underwater vehicles, or AUVs, has created a new generation of deep-sea observation platforms that combine the advantages of the EITS with the ability to move through the water column. AUVs are untethered, self-propelled vehicles that can be programmed to follow specific transects, survey designated areas, or hover at specific depths for extended periods. Modern AUVs can be equipped with bioluminescence sensitive cameras, bathyphotometers that measure ambient light levels at specific wavelengths, and hydrophones that record the acoustic environment simultaneously with visual observations. They are quieter than ROVs, produce less water turbulence, and can operate independently for hours or even days before being recovered. Some AUVs have been specifically designed for bioluminescence research, equipped with ultra-sensitive photomultiplier tubes capable of detecting single photons in the deep-sea environment, allowing them to map the three-dimensional distribution of bioluminescent organisms across vast volumes of water in ways that would be impossible with a fixed camera or a short-duration ROV dive. One of the most significant technological developments in deep-sea bioluminescence research has been the deployment of permanent seafloor observatories. The Monterey Accelerated Research System, or MARS, operated by Embari in Monterey Bay, provides continuous, real-time data from the deep ocean floor. Connected by fiber optic cable to shore-based facilities, MARS can supply power and data bandwidth to a variety of instruments deployed on the seafloor at a depth of approximately 900 meters. This includes cameras, current meters, chemical sensors, and bioluminescence detectors. The significance of MARS is that it provides continuous observation, not a snapshot, not a brief visit by an ROV that lasts a few hours and then departs. Continuous, uninterrupted monitoring of the deep ocean, day and night, week after week, month after month. The data returned by MARS and similar observatories, including Ocean Networks Canada's Neptune Observatory in the Northeast Pacific and the European Multidisciplinary Seafloor and Water Column Observatory, or EMSO, have revealed temporal patterns in deep-sea bioluminescence that would have been completely invisible to snapshot observation methods. Bioluminescent activity varies with the seasons, it varies with lunar cycles, it varies with tidal cycles. It varies in ways that do not correspond to any known environmental variable, and for which no explanation has been proposed. The hydrothermal vents discovered along mid-ocean ridges present their own peculiar bioluminescent puzzle. These vents, first observed by humans in 1977 near the Galapagos Rift, emit superheated water laden with dissolved minerals at temperatures exceeding 300 degrees Celsius. The vent ecosystems are powered not by sunlight, but by chemosynthetic bacteria that derive energy from the chemical compounds in the vent fluid. The ecosystems that surround hydrothermal vents, the giant tube worms, the vent crabs, the shrimp, the dense bacterial mats, are among the most remarkable biological communities on Earth, thriving in conditions of extreme heat, extreme pressure, extreme chemistry, and total darkness. Some organisms associated with hydrothermal vent communities have been observed to produce bioluminescence. This is puzzling because the environmental conditions near vents are so extreme, and the energy budgets of vent organisms are so tightly constrained by the chemistry of their environment that the additional metabolic cost of bioluminescence seems difficult to justify. In an ecosystem where every calorie is derived from sulfur chemistry rather than sunlight, the luxury of producing light for communication seems extravagant. And yet it occurs. Why organisms, in one of the most energy-constrained environments on Earth, invest metabolic resources in producing light is a question that remains open. Equally mysterious are the brine pools found in certain deep-sea basins, particularly in the Gulf of Mexico and the Red Sea. These are lakes of hypersaline water that sit on the ocean floor, so dense with dissolved salt that they form a distinct layer beneath the overlying seawater, complete with their own shoreline, their own surface, and their own wave action. The salinity of brine pools is so extreme that most organisms that venture into them are killed almost instantly. The borders of brine pools are often littered with the remains of fish, crabs, and other organisms that swam too close and were overwhelmed by the toxic chemistry. And yet, at the edges of some brine pools, researchers have observed bioluminescent displays. Flashes and glows emanating from the transition zone where the brine meets the surrounding seawater. Some of these may be produced by organisms in the process of dying, their bioluminescent systems firing as their cellular machinery breaks down, but others appear sustained and purposeful, suggesting the presence of extremophilic organisms that have adapted to survive at the interface between the brine and the surrounding water, and that use bioluminescence for reasons that remain completely unknown. The phenomenon of milky seas represents perhaps the most spectacular and least understood manifestation of bioluminescence on the planet. Milky seas are vast areas of the ocean surface that glow with a uniform, steady, whitish-blue light visible to the naked eye, and in extreme cases, visible from space. Historical accounts of milky seas date back centuries. Sailors in the Indian Ocean and the Arabian Sea have reported encountering patches of luminous water extending from horizon to horizon, turning the sea into what one account described as a field of snow. The glow is not the brief, responsive flash of typical bioluminescence. It is a continuous, steady emission that can persist for hours or even days, covering areas that satellite imagery has measured at more than 15,000 square kilometers. The leading hypothesis attributes milky seas to massive blooms of bioluminescent bacteria, specifically species of Vibrio harveyi or related organisms that colonize the surface of algal blooms or organic debris in concentrations sufficient to produce visible light across an enormous area. But the mechanism that triggers such coordinated, simultaneous luminescence across thousands of square kilometers of ocean surface remains poorly understood. Bacteria produce bioluminescence through a process called quorum sensing, in which individual cells begin producing light only when the density of bacteria in their immediate surroundings reaches a critical threshold. The signal that initiates light production is a chemical molecule produced by each individual bacterium. When enough bacteria are present, the concentration of the signaling molecule crosses a threshold, and the entire population begins glowing simultaneously. For a milky sea to form, the bacterial population must reach quorum across an area larger than some countries. The logistics of this process, the conditions required to sustain it, and the frequency with which it occurs are all poorly constrained. Satellite observations have confirmed at least one milky sea event in the Indian Ocean in 2005, validating centuries of sailor testimony that had been dismissed by many scientists as exaggeration or folklore. The story of bioluminescence in Japanese culture offers a different lens through which to view these phenomena. The sea firefly, Vagula hilgendorfi, known in Japanese as umi hotaru, is a tiny ostracod crustacean found in shallow coastal waters around Japan. When disturbed, it releases a cloud of luminous blue mucus into the water, creating a glowing trail that can persist for several seconds. Umi hotaru have been known to Japanese coastal communities for centuries and hold a place in the cultural imagination that goes beyond scientific curiosity. They appear in poetry. They are collected by children on summer evenings. Coastal fishermen have used their glow as a form of ambient entertainment during long nights at sea. The Umi hotaru represents something that Western science has often struggled to capture in its papers and datasets. The sense of wonder, of beauty, of deep aesthetic response that bioluminescence evokes in the human observer. Science can explain the chemistry. It can identify the species. It can measure the wavelength and duration and intensity of the glow. But it cannot, in its formal language, capture the experience of standing on a Japanese shore at night and watching the water light up with 1,000 pinpoints of blue, each one a tiny organism spending its metabolic resources on a display of light that no human was meant to see, that serves no human purpose, and that existed for millions of years before human eyes evolved to appreciate it. During the Second World War, Japanese soldiers reportedly collected dried umi hotaru and used their bioluminescent properties to read maps and messages at night without revealing their position to enemy forces. The dried ostracods, when moistened with water, produced a faint blue glow sufficient to illuminate text held close to the hand but too dim to be visible at any significant distance. Whether this practice was widespread or merely anecdotal is debated, but the story illustrates a fundamental truth about bioluminescence that extends far beyond military applications. Humans have been aware of biological light for as long as they have gone to sea. They have been awed by it, frightened by it, inspired by it, and occasionally put it to practical use. But they have never fully understood it. The gap between human awareness of bioluminescence and human understanding of it is measured in centuries. We notice the light almost immediately. We are still, after all this time, trying to figure out what it means. The barrel-eye fish, Macropinna microstoma, represents one of the most extraordinary adaptations to the bioluminescent environment of the deep ocean. This small fish, found at depths between 200 and 800 meters in the North Pacific, has a completely transparent head. Its skull is a fluid-filled dome of clear tissue through which its internal structures, including its brain, are visible. Its eyes are large, tubular, and pointed upward, positioned inside the transparent dome of its head. These tubular eyes are capable of rotating within the dome, scanning the water above for the silhouettes of prey against the faint light filtering down from the surface. The transparent head allows the eyes to look in directions that the rigid structure of a normal fish skull would block. The barrel eye is, in effect, looking through its own head. It can look straight up, detecting the faintest shadows of potential prey against the dim light of the mesopelagic zone, and it can rotate its eyes forward to look ahead when it moves to strike. The transparent dome also contains two small organs above the mouth that were initially mistaken for eyes, but are actually olfactory structures. The fish has evolved a head that is simultaneously a window, a periscope, and a sensory platform, all in service of navigating a world where the dominant form of information is light, whether that light comes from the surface above, the bioluminescent signals of nearby organisms, or the counter-illumination of prey attempting to hide against the faint glow of the water column. The barrel eye was first described in 1939, but it was not until 2004 that researchers at MBARI captured the first footage of a living barrel eye in its natural habitat using an ROV in Monterey Canyon. The footage revealed for the first time that the transparent dome and the rotatable eyes were not merely structural curiosities but active, functional adaptations that the fish used continuously as it hovered in the water column, scanning its environment for the faintest bioluminescent cues. Before this footage, the barrel eye had been known only from specimens brought to the surface by trawl nets, specimens in which the transparent dome invariably collapsed and the eyes were frozen in a fixed position. The living animal was utterly different from the dead one. It was dynamic, alert, actively engaged in reading the luminous environment around it. The dead specimens in museum jars had told scientists almost nothing about how this animal actually lived. The deep ocean had to be visited on its own terms for the truth to emerge. What the barrel eye illustrates, more vividly than almost any other organism in the deep ocean, is how thoroughly bioluminescence has shaped the evolution of deep-sea life. Eyes like the barrel eye's tubular organs did not evolve to detect sunlight. They evolved to detect bioluminescence, to read the signals, to interpret the flashes, the glows, the counter-illumination patterns. The barrel eye's extraordinary anatomy is a direct evolutionary response to the bioluminescent environment in which it lives. Its transparent head, its rotatable eyes, its ability to detect the faintest photon against a background of absolute darkness. These are not random adaptations. They are specific solutions to specific problems posed by a world in which the primary medium of information is biological light. And if the barrel eye has evolved this degree of specialization to read bioluminescent signals, the question becomes unavoidable. How many other organisms in the deep ocean have evolved equally remarkable, equally specialized, equally extraordinary adaptations to the luminous world around them that we have never seen, never examined, and never imagined? Because we have never been able to observe them without disturbing them, blinding them, or destroying them in the process of trying to look. And it gets worse, because the individual signatures of bioluminescent output are beginning to reveal a layer of complexity that no one anticipated. Research on certain species of black dragonfish has shown that individual organisms produce bioluminescent patterns that differ from one individual to the next. Not species-level patterns, individual level patterns, variations in flash rate, intensity, spectral distribution, and spatial arrangement of photophores that are as unique to each individual dragonfish as a fingerprint is to each individual human. If these variations are meaningful, if they carry information about identity, fitness, size, sex, reproductive status, or territorial boundaries, then bioluminescence in the deep ocean is not merely a language. It is a language with accents, a language with dialects, a language in which every speaker has a voice that is recognizably their own. The implications of individual bioluminescent signatures would transform the field. It would mean that deep-sea organisms are not merely broadcasting generic signals to anonymous receivers. They are engaged in individual recognition. They know, or at least can determine, who is flashing at them. The conversation is personal, and we cannot read it. We cannot even see it clearly enough to determine whether it is happening. The evidence is suggestive, tantalizing, and profoundly incomplete, trapped in the same darkness that has hidden these organisms from human understanding since the beginning. Consider the fundamental challenge this poses. If individual bioluminescent signatures exist, then studying bioluminescence at the species level, which is how it has been studied for the past century, is like studying human language by recording only vowel sounds. The species-level patterns are the broad categories, the vowels. The individual signatures are the consonants, the syntax, the grammar, the meaning. Without them, the language is noise. With them, the noise is conversation. And the technology required to detect, record, and analyze individual-level bioluminescent signatures in the deep ocean across multiple organisms in real time over extended periods does not yet exist. Scientists can measure the bioluminescence of individual organisms captured and brought to the surface, but capturing them alters their physiology, stresses their systems, and almost certainly changes their bioluminescent output. The organism in the laboratory is not the organism in the ocean. The flash measured in a darkened tank aboard a research vessel is not the flash produced in the darkness of the bathypelagic zone at 2,000 meters depth, under 500 atmospheres of pressure, in water near freezing. The context is different, and in bioluminescence, as in all language, context is everything. The siphonophores, colonial organisms related to jellyfish, display coordinated bioluminescent patterns that suggest a level of collective behavior unlike anything else in the animal kingdom. A siphonophore is not a single organism. It is a colony of individual zooids, each specialized for a particular function: feeding, reproduction, locomotion, defense, all connected in a chain that can extend for tens of meters, making some siphonophores the longest animals on Earth. When a siphonophore colony produces bioluminescence, the display can propagate along the entire length of the colony in coordinated waves, rippling from one end to the other in sequences that vary in speed, direction, and intensity. Different parts of the colony can produce different colors or different flash patterns simultaneously. The colony behaves bioluminescently as a single coordinated entity while being composed of thousands of individual organisms. How this coordination is achieved, how the signal to luminesce is transmitted from one zooid to the next across the length of the colony, and what information, if any, is encoded in the variations of the display, are questions that remain almost entirely unanswered. The siphonophore's bioluminescent display is a collective performance, a symphony played by an orchestra in which every musician is technically a separate organism, but all play in concert, and no one has yet written down the score. In 2020, researchers from the Schmidt Ocean Institute, aboard the research vessel Falkor, filmed a siphonophore in the deep waters off the coast of Western Australia that measured approximately 47 meters in length. 47 meters, longer than a blue whale, longer than most ships. The organism was coiled in a vast spiral, its individual zooids pulsing with faint bioluminescence that travelled in waves along the enormous chain. The footage, captured by the ROV Sebastian, was shared widely on social media and in scientific publications. But what it represented was not merely a large organism. It represented a colony of thousands of individual living things, each one capable of producing light, all doing so in coordination, in patterns, in what appeared to be a structured and purposeful display. The siphonophore was glowing, it was speaking, and everything it said was incomprehensible to the humans watching through the camera. There is a term in deep-sea biology that has begun to appear with increasing frequency in the scientific literature. The term is dark biodiversity. It refers to the organisms, species, and biological processes that exist in the deep ocean but have never been observed, never been documented, and never been included in any census or estimate. Dark biodiversity is not a metaphor. It is a measurement gap. It is the difference between what exists and what we know exists. And every technological advance in deep-sea observation has revealed that dark biodiversity is larger than anyone expected. The deep ocean does not become simpler as we look more closely. It becomes more intricate, more layered, more densely populated, and more luminous. The darkness is full of light, the silence is full of conversation, the emptiness is teeming with life, and everything we think we know about it is based on glimpses through keyholes, moments in time, slivers of space, fragments of a picture so vast that even the fragments are overwhelming. The irony is not lost on the researchers who study it. Humanity has mapped the surface of Mars in greater detail than the floor of its own ocean. It has landed vehicles on the Moon and on asteroids, has sent probes beyond the edge of the solar system, has imaged black holes in distant galaxies, and measured the chemical composition of exoplanet atmospheres light years away. But it has not yet managed to observe, for more than a few fleeting hours at a time, what is happening in the darkness less than 2 miles beneath the surface of its own planet. The deep ocean is closer than the Moon. It is infinitely more accessible than Mars. It contains more life than any other environment on Earth, and it is, by almost any meaningful measure, less understood than the surface of a world more than 50 million kilometers away. The reason is not a lack of interest. It is not a lack of funding, though funding for deep-sea research is perennially modest compared to the budgets of space agencies. The reason is that the deep ocean is, in purely physical terms, one of the most hostile environments that human technology has ever attempted to penetrate. The pressure alone would crush any unprotected vehicle. The temperature hovers near freezing. The darkness is absolute, and the distances involved, not the vertical distance from the surface, which is comparatively manageable, but the horizontal distances, the sheer lateral extent of the ocean basins, are so vast that comprehensive survey is not merely difficult. It is, with current technology, impossible. What all these explorations and innovations have collectively revealed is a picture that grows more complex with every expedition, every camera deployment, every new technology brought to bear on the problem. The deep ocean is not yielding to understanding. It is deepening the mystery with every answer it provides. And the researchers who have devoted their careers to this work, the Widders, the Haddocks, the Johnsons, the countless graduate students and technicians and ROV pilots who have spent their lives trying to see what the deep ocean is doing in the dark, all share the same humbling recognition. They are looking through a keyhole at a cathedral. They can see fragments of the architecture, glimpses of the stained glass, echoes of the music, but the full structure, the complete picture, the entire scope of what the deep ocean is doing with its light remains as far beyond their current understanding as the cathedral is beyond the keyhole. The instruments are improving, the windows are widening, but the cathedral keeps getting larger. Something is wrong with our assumptions. For centuries, human beings have treated the deep ocean as an absence of light, an absence of warmth, an absence of life, an absence of everything that makes a place habitable, interesting, or worthy of attention. The deep ocean, in the human imagination, has been defined by what it lacks rather than by what it contains. It is dark, it is cold, it is empty, it is hostile, it is a wasteland, a void, a negative space on the map of the world. The surface ocean is teeming, colourful, productive. The deep ocean is its opposite. The basement of the planet, the place where things sink and are forgotten. This assumption is wrong, not merely incomplete, not merely oversimplified. It is fundamentally, categorically, demonstrably wrong. And the evidence that proves it wrong is glowing, in the darkness, across every ocean basin on Earth, in quantities, and with a consistency that should have overturned the assumption decades ago. The deep ocean is not empty. It is the most populated environment on Earth. It is not dark in any biologically meaningful sense. It is ablaze with self-generated light. It is not silent. It is having conversations that we cannot hear and can barely detect. It is not a wasteland. It is the default biome of the planet, the largest habitable volume on Earth, the environment in which the majority of all multicellular life exists. We did not discover life in the deep ocean. Life was always there. We are the latecomers. We are the anomaly. We are the organisms who evolved on a thin, sunlit film on the surface of a planet whose true character is defined by pressure, darkness, and biological light. Let that sink in. The surface of the Earth, the thin layer of atmosphere, land and sunlit water in which human beings live, in which all of human history has taken place, in which every civilization has risen and fallen, in which every ecosystem that we consider normal and familiar operates, that layer represents a tiny fraction of the habitable volume of the planet. The deep ocean, by contrast, represents more than 90% of the living space on Earth. More than 90%. If you were to select a random point in the habitable biosphere of this planet, a random cubic meter of space in which an organism could theoretically live, the overwhelming probability is that you would select a point in the deep ocean. Not on land, not in the atmosphere, not in a forest or a desert or a meadow or a river. In the deep ocean, in the dark, under pressure, where the primary form of communication is light generated by living chemistry. That is the normal condition of life on Earth. That is what a typical organism on this planet experiences. Sunlight, open air, moderate pressure, visible landscapes, these are the exception. Darkness, cold, crushing pressure, and bioluminescence are the rule. The numbers reinforce this inversion. The ocean covers approximately 71% of the Earth's surface, and its average depth is approximately 3,700 meters, well within the range of total darkness. The total volume of the deep ocean below the photic zone is approximately 1.3 billion cubic kilometers. 1.3 billion. The total volume of all freshwater lakes, rivers, and streams on Earth is approximately 105,000 cubic kilometers. The entire atmosphere of the planet, from the surface to the edge of space, contains less habitable volume than the deep ocean alone. In volumetric terms, the deep ocean dwarfs every other environment on Earth combined. It is not a minor habitat, it is not a marginal zone. It is the dominant feature of the planet's biosphere, and the organisms that live in it, the bioluminescent organisms that glow and flash and pulse in its darkness, are not fringe species. They are the majority. They are what life on Earth mostly looks like. This realization demands a philosophical reckoning that science has been slow to undertake. When we speak of life on Earth, we almost always mean life as we experience it. Terrestrial life, life in the light, life under atmospheric pressure, breathing gaseous oxygen, navigating by vision in an illuminated environment. But this is not what life on Earth mostly is. Life on Earth is mostly aquatic. It is mostly dark. It is mostly under pressure. It is mostly bioluminescent. The word normal, when applied to life on this planet, should not evoke a sunlit meadow or a tropical forest. It should evoke a column of black water, 3,000 meters deep, at near freezing temperature, under hundreds of atmospheres of pressure, flickering with flashes of blue and green light produced by organisms that have never seen the sun and never will. That is normal. That is the baseline. That is what life on Earth actually looks like when you step back far enough to see the whole picture, rather than just the tiny, brightly lit corner in which you happen to stand. The metaphorical inversion extends even further. Humans have always used light as a symbol of knowledge, understanding, and truth. We speak of enlightenment. We speak of illumination. We speak of bringing things to light, of seeing the light, of emerging from darkness into understanding. Darkness, in the human symbolic vocabulary, represents ignorance, fear, evil, the unknown. But in the deep ocean, this metaphor collapses. In the deep ocean, darkness is not the absence of knowledge. It is the medium in which the most complex biological conversations on Earth take place. Darkness is not ignorance. It is the environment that gave rise to the most sophisticated use of light that evolution has ever produced. Darkness is not the obstacle to understanding. It is the canvas on which life has painted its most intricate, most beautiful, and most enduring work. The organisms of the deep ocean did not overcome the darkness. They embraced it. They made it their own. They turned the absence of sunlight into the presence of something far more nuanced, far more variable, far more information-rich than sunlight ever was. Sunlight is constant, uniform, external. Bioluminescence is controlled, variable, personal. It carries meaning that sunlight cannot carry. It can be turned on and off, varied in intensity, modulated in wavelength, directed at specific recipients, shaped by evolution to convey specific messages. The organisms of the deep ocean have not merely adapted to darkness, they have transcended it. They have built a civilization of light that needs no sun. Consider what it would mean if we found this on another world. If a space probe descended into the lightless ocean of Europa, the ice-covered moon of Jupiter, and found there a world teeming with organisms that communicated in self-generated light, that had evolved dozens of independent chemical pathways for producing photons, that used light for hunting, defense, mating, navigation, and forms of communication that the probe's instruments could detect but not decode, it would be the greatest scientific discovery in human history. It would rewrite every textbook, it would dominate every headline, it would reshape humanity's understanding of its place in the universe. And yet this is exactly what exists on Earth, right now, in the deep ocean, in a world that is, in many respects, as alien and as unknown as the subsurface oceans of distant moons. The difference is proximity. We cannot reach Europa. We can reach the deep ocean. And yet, paradoxically, we know more about the chemical composition of Europa's ice shell, measured by spectroscopy from spacecraft millions of kilometers away, than we know about the chemical ecology of bioluminescent organisms 3 kilometers beneath the surface of our own ocean. The comparison between deep-sea exploration and space exploration is not merely rhetorical. It illuminates a genuine imbalance in human curiosity, or at least in the allocation of resources to satisfy that curiosity. 12 human beings have walked on the surface of the moon. Fewer than a dozen have visited the Hadal Zone, the deepest region of the ocean. The total number of hours that human eyes have directly observed the deep ocean through the windows of manned submersibles is a fraction of the time that humans have spent in orbit around the Earth. Space agencies around the world collectively spend tens of billions of dollars annually searching for evidence of microbial life on Mars, a search that has so far found nothing conclusive. Meanwhile, a world of trillions of bioluminescent organisms, many of them unknown to science, many of them producing light through chemical pathways that have never been characterized, exists within reach of any vessel capable of lowering a camera on a cable. The deep ocean does not require rockets. It does not require escape velocity. It does not require years of transit through the vacuum of space. It requires a ship, a cable, and a camera that can survive the pressure. And yet, proportionally, humanity has invested far more in looking outward than in looking downward. We are a species that searches the sky for signs of life while standing on an ocean full of it. The technology that is slowly widening our window into this world is itself undergoing a transformation. The Monterey Accelerated Research System, MARS, represents one paradigm, a permanent cabled observatory connected to shore-based facilities, providing continuous, real-time data from the deep ocean floor. Ocean Networks Canada's Neptune Observatory represents another iteration of the same concept, a fiber optic network spanning the Juan de Fuca tectonic plate, with nodes at various depths, each equipped with cameras, sensors, and data transmission capabilities that allow researchers anywhere in the world to observe the deep ocean in real time through a web browser. The European Multidisciplinary Seafloor and Water Column Observatory, EMSO, is building a similar network across multiple sites in European waters. These observatories are not visits, they are presences. They represent a fundamentally different relationship between human science and the deep ocean, one based not on brief expeditions, but on sustained attention. And the data they have returned have already begun to reveal patterns that no expedition could have detected. Bioluminescent activity at the Mars site varies with the seasons. It peaks in autumn, when the flux of marine snow, the constant rain of organic particles sinking from the productive surface waters above, reaches its maximum. It dips in spring, when surface productivity is high, but the particles have not yet had time to sink to observatory depth. It fluctuates with storm events on the surface, which stir up the water column and disrupt the normal patterns of vertical migration. It responds to deep water current shifts, to changes in temperature at depth, to variations in dissolved oxygen concentration. The deep ocean is not static. It has rhythms. It has seasons. It has moods, if such a word can be applied to a body of water, and its bioluminescence tracks those rhythms with a fidelity that suggests the luminous organisms of the deep are intimately connected to processes occurring hundreds or thousands of meters above them, in a world they will never visit. The deep ocean is not isolated from the surface. It is linked to it by the invisible threads of physics, chemistry, and biology, and the bioluminescent activity at depth is in some sense a luminous echo of events on the surface. A storm churns the water above, and days or weeks later, the organisms at depth respond with changes in their patterns of light. A phytoplankton bloom feeds the surface food web, and months later, the rain of organic particles reaching the deep sea floor intensifies, and the organisms there glow brighter, more frequently, in patterns that change with the changing food supply. And then there are the observations that fit no pattern at all, unexplained surges of bioluminescent activity that do not correlate with any measured environmental variable. Events lasting hours or days in which the background level of luminescence at a monitoring site increases by an order of magnitude and then returns to baseline with no apparent cause. Deep-sea cameras recording what appears to be coordinated bioluminescent activity across areas larger than the camera's field of view, suggesting that whatever is producing the light is communicating across distances of tens or hundreds of meters in patterns that no model predicts and no theory explains. These events are not artifacts. They are too consistent, too well documented, recorded on too many instruments by too many independent observers to be dismissed as equipment malfunction or observer error. They are real, they are happening, and they are, as of now, unexplained. There are moments when the data from these observatories suggests something that researchers are reluctant to say aloud because saying it aloud would imply a degree of organized complexity that current models of deep-sea ecology are not equipped to accommodate. The coordinated bioluminescent events, the unexplained surges, the patterns that propagate across distances too large for any single organism to span. These observations are consistent with the hypothesis that deep-sea bioluminescence is not merely a collection of individual organisms producing individual signals. It may be, at least in some instances, a network phenomenon, a collective behavior emerging from the interactions of thousands or millions of organisms, each one producing its own small contribution to a larger pattern, a pattern that none of them individually controls, but all of them collectively create. Emergence, self-organization, the spontaneous appearance of order from the uncoordinated actions of many simple agents. These are concepts well studied in other fields. Ant colonies exhibit emergent behavior, flocking birds exhibit self-organization, but the possibility that the bioluminescent displays of the deep ocean might exhibit the same kind of emergent, self-organized, collectively intelligent behavior is an idea that lies at the outermost edge of current scientific speculation. It is not established, it is not proven, it is barely even hypothesized because the data are too sparse and the observations too fragmentary to test it rigorously. But the hints are there, in the data from the observatories, in the footage from the ROVs, in the anomalous surges, and the unexplained coordinations and the patterns that propagate through the darkness, as if something, or many things, were passing a message along a chain that stretches beyond the camera's view. If these hints are confirmed, if future observations establish that deep-sea bioluminescence does indeed exhibit emergent, self-organized behavior at scales larger than any individual organism, the implications would extend far beyond marine biology. It would mean that the deep ocean contains a form of collective information processing that has no parallel in any known terrestrial ecosystem, not merely communication between individuals, which is common throughout the animal kingdom, but a form of collective signaling in which the behavior of the group produces patterns that are more complex, more information-rich, and more organized than the behavior of any individual member. It would mean that the deep ocean is not just glowing, It is computing, processing information, responding collectively to stimuli, generating patterns that carry meaning at a scale that transcends the individual and operates at the level of the community, the population, or even the ecosystem. This idea is speculative, it is unproven, but it is not impossible, and the simple fact that it cannot be ruled out, that the data permit it, even if they do not yet support it, is itself a statement about how little we understand about what is happening in the darkness below. Consider the abyssal worms that have been observed producing sequential bioluminescent flashes along the length of their bodies, creating what one researcher described as a runway of lights. The flashes propagate from head to tail in evenly spaced intervals, like the landing lights of an airport runway, each flash illuminating for a fraction of a second before the next one in sequence ignites. The pattern repeats. It cycles. It runs from one end of the worm to the other and then starts again. No explanation for this behavior has been proposed that satisfies the scientific community. It is not a defensive display, because the worm continues the pattern regardless of whether a threat is present. It is not an obvious mating signal, because the worm produces the pattern when alone. It does not appear to serve a feeding function. It is a pattern of light, produced at metabolic cost, with no identified purpose, repeating in the darkness at the bottom of the ocean, where, as far as current science can determine, nothing is watching, and yet it continues. The worm lights its runway, flash by flash, in the blackness, for an audience of none, or for an audience that we have not yet found. The philosophical implications of what we are beginning to learn about deep-sea bioluminescence extend beyond biology, beyond ecology, beyond even the philosophy of science itself. They touch on something more fundamental. They touch on the question of what it means to know a planet. Humanity has mapped the Earth's surface in extraordinary detail. Satellite imagery covers every square meter of land. Sonar surveys have mapped the topography of the ocean floor at resolutions of a few kilometers, and in some areas at resolutions of meters. We know where the mountains are. We know where the trenches are. We know the temperature of every sea surface and the height of every wave. But we do not know what is happening in the water column between the surface and the bottom. We do not know what lives there. We do not know what it is doing. We do not know what it is saying. The deep ocean is the largest blank space on the map of life on Earth, and it is not blank because nothing is there. It is blank because we have not looked. We have not been able to look. And when we do look briefly through the narrow windows of our ROVs and submersibles and observatory cameras, what we see is a world so rich, so complex, so thoroughly and densely inhabited by organisms doing things we cannot explain, that the glimpse is more humbling than the ignorance. Let that sink in. 95% of the ocean remains unexplored. Not 95% of the deep ocean, not 95% of the abyssal zone, 95% of the entire ocean. The vast majority of the single largest habitat on the planet has never been visited, never been surveyed, never been observed by any human instrument. The 5% that has been explored, the tiny fraction from which all of our knowledge of deep-sea bioluminescence is drawn, has already revealed more than 50 distinct bioluminescent strategies, more than 700 documented bioluminescent species, more than 40 independent chemical origins of light production, and an uncounted number of bioluminescent behaviors that have been observed but never explained. If the explored 5% contains this much complexity, this much diversity, this much mystery, then the unexplored 95% contains something that current science cannot quantify and current imagination cannot encompass. This is not science fiction. This is the current state of scientific knowledge about the planet we live on. The single most biologically productive, most densely inhabited, most biochemically diverse environment on Earth is one that we have barely begun to investigate. The bioluminescent world beneath 3,000 feet is not a footnote in the story of life on this planet. It is the main text, the story of terrestrial life, the story that humans know best and tell most often, the story of forests and grasslands and deserts and rivers and the organisms that inhabit them. That story is the footnote. It is the exception. It is the small, sunlit, atypical corner of a planet that is, for the most part, dark, pressurized, aquatic, and glowing with biological light. The question of what this means for humanity's self-understanding is one that science alone cannot answer. It is a philosophical question, an existential one. Human beings have always placed themselves at the center of the natural world. The environments we know best, the environments we have shaped and inhabited, the environments in which our cultures and civilizations have developed. These are the environments we consider normal, default, representative. We call them nature. We call them the natural world, and we treat everything else, the deep ocean, the upper atmosphere, the subsurface of the earth, the interior of glaciers, as extremes, as anomalies, as places that are defined by their deviation from the norm. But the deep ocean is not an anomaly. The deep ocean is the norm. It is the largest habitable environment on the planet. It contains the majority of the planet's living organisms. It hosts the most widespread form of communication on Earth, bioluminescence, and it has existed in essentially its current form for longer than any terrestrial biome. The forests that we consider ancient have existed for perhaps 350 million years, according to current geological estimates. Grasslands are younger, deserts shift with the climate, but the deep ocean has been dark, cold, pressurized, and inhabited for as long as there has been an ocean at all. It is the oldest continuously inhabited environment on the planet. The bioluminescent organisms that live in it are inheritors of a tradition of biological light production that stretches back through an expanse of time so vast that the human mind can acknowledge it but cannot feel it. What this means, practically, is that everything we think we know about life on Earth is derived from a biased sample. We have studied the exception and assumed it was the rule. We have described the surface and assumed it was representative. We have catalogued the organisms of the sunlit world and assumed they constituted the majority. They do not. The majority of life on this planet lives in the dark. The majority of organisms on this planet can make their own light. The majority of biological communications on this planet is conducted in photons rather than sound waves or chemical signals. If we were to write an honest natural history of this planet, one that weighted each environment according to its volume, its biomass, and its biological diversity, rather than according to human familiarity, the deep ocean would dominate every chapter. It would be the setting for the main narrative. The surface world, the world of sunlight and air and moderate pressure, would appear as an unusual, specialized, and in many ways impoverished outlier, a bright anomaly on the surface of a dark planet. The implications of this perspective shift are not merely academic. They reshape every question we ask about life on Earth. When biologists ask what is normal for life on this planet, the answer is darkness, pressure, cold, and bioluminescence. When ecologists ask what the dominant communication system of the biosphere is, the answer is light, self-generated, chemically produced, evolutionarily refined across dozens of independent lineages. When philosophers ask what defines the character of this planet as a living world, the answer is not photosynthesis, not terrestrial ecosystems, not the oxygen-rich atmosphere. The answer is the deep ocean. The answer is the glow. The answer has been glowing beneath us for longer than we have known how to ask the question, and we are only now, in the most recent instant of geological time, beginning to realize that the answer was there all along. Beneath the surface, beneath the light, in the darkness where the real light lives. And yet, even as we begin to reckon with this inversion, the deep ocean is changing. Ocean warming is altering the temperature structure of the water column, shifting the boundaries between oceanic zones, changing the depth at which organisms find their optimal conditions. Ocean acidification, caused by the absorption of carbon dioxide from the atmosphere, is changing the chemistry of seawater in ways that may affect the biochemical reactions that produce bioluminescence. Deoxygenation, the expansion of low oxygen zones in the deep ocean, is reducing the habitable volume available to aerobic organisms, including many bioluminescent species. The deep ocean, despite its remoteness, despite its depth, despite its apparent isolation from the surface world, is not immune to the changes that human activity is imposing on the planet's climate and chemistry. The bioluminescent world beneath 3,000 feet, The world that has glowed in the darkness since before any human civilization existed is being altered by forces originating on the surface, in the atmosphere, in the industrial processes and energy systems of a species that has barely begun to understand what it is changing. There is a deep irony here that deserves to be named. We are changing the deep ocean before we understand it. We are altering its chemistry before we have catalogued its chemistry. We are warming its waters before we know what lives in them. We are reducing its oxygen before we have measured how much oxygen its organisms need. The bioluminescent conversations of the abyss may be changing in response to these pressures, shifting in intensity, in wavelength, in frequency, in meaning. And we would not know because we have not yet established the baseline against which change can be measured. We are editing a book we have not read. We are revising a manuscript we have never opened. We are changing the rules of a game whose rules we have not learned. And the organisms whose light has sustained the most extensive communication network on this planet for an unimaginable span of time are experiencing the consequences of our actions in ways that we may never be able to observe, let alone reverse. The deep ocean does not need us to understand it. It has functioned without our understanding for longer than we have existed as a species. The bioluminescent conversations of the abyss predate the emergence of the human brain by an immensity of time that words can gesture toward but never truly convey. They will continue after every human institution, every human language, every human technology has been forgotten. The light in the deep ocean is not waiting for us to decode it. It is not performing for our cameras. It is not cooperating with our research agendas. It is simply doing what it has always done, carrying information through darkness, connecting organisms across the void, sustaining a web of communication so vast and so ancient that our most sophisticated instruments can barely detect its edges. And the deeper truth, the truth that the deep ocean has been patiently, luminously demonstrating for anyone willing to descend far enough to see it, is that life does not need the sun. Life does not need the surface. Life does not need warmth, or moderate pressure, or breathable atmosphere, or any of the conditions that human beings consider essential. What life needs, all that life needs, is chemistry, darkness, and time. Given those three ingredients, life will find a way to make its own light, and it will use that light to build something magnificent. The bioluminescent world of the deep ocean has already given humanity more than wonder. It has given us tools. The discovery of green fluorescent protein, or GFP, in the jellyfish Aquaria victoria by Osamu Shimomura in the 1960s set in motion a chain of scientific developments that would reshape the landscape of modern biology. Shimomura first isolated GFP while studying bioluminescence at the Friday Harbor Laboratories in Washington state, extracting the protein from thousands of individual jellyfish collected from the cold waters of Puget Sound. The protein glowed green when exposed to ultraviolet light, a property that seemed, at the time, to be a scientific curiosity and little more. It took decades for the full potential of GFP to become apparent. In the 1990s, researchers Martin Shalfie and Roger Sien demonstrated that the gene for GFP could be inserted into other organisms, turning it into a biological tag, a glowing marker that could be attached to specific proteins, specific cells, specific structures within living tissue, allowing scientists to track biological processes in real time under a microscope. The protein glowed, and wherever it went, whatever it was attached to, it made the invisible visible. In 2008, Shimomura, Shalfie, and Sien were awarded the Nobel Prize in Chemistry for the discovery and development of green fluorescent protein. The Nobel Committee described GFP as one of the most important tools in contemporary bioscience. It had transformed the study of cell biology, genetics, developmental biology, neuroscience, and medicine. Researchers used GFP variants to visualize the growth of cancer cells in living tissue. They used it to track the development of nerve connections in embryos. They used it to monitor the spread of infections in real time. They created variants that glowed in different colors: blue, cyan, yellow, red, allowing multiple processes to be tracked simultaneously within the same organism. The deep ocean, through a single jellyfish collected from shallow water had provided the key to seeing what had previously been invisible inside living cells. A protein that a marine organism produced for reasons related to its own bioluminescent signaling, reasons that are still not entirely understood, had become one of the foundational tools of modern molecular biology. But the problem is this: GFP is one protein from one species discovered by one researcher more than half a century ago. The deep ocean contains hundreds, perhaps thousands, of bioluminescent proteins, in hundreds, perhaps thousands, of species, the vast majority of which have never been characterized. If a single protein from a single shallow water jellyfish could generate a Nobel Prize and transform multiple fields of biology, what might the biochemical libraries of the deep ocean contain? What enzymes, what proteins, what chemical pathways are locked inside organisms that have never been collected, never been cultured, never been subjected to the analytical techniques that could reveal their properties? The potential is not theoretical, it is chemical, it is molecular. It is waiting in the darkness at the bottom of the ocean for someone to develop the technology to access it without destroying it in the process. The efficiency of bioluminescent reactions offers another profound implication for human technology. A standard incandescent light bulb converts approximately 5% of its electrical energy into visible light. The remaining 95% is wasted as heat. Even modern LED technology, while far more efficient, still loses a significant fraction of its energy to thermal dissipation. Bioluminescent reactions, by contrast, convert approximately 90% of their chemical energy into light. 90%. They are among the most efficient light-producing reactions known to chemistry. They produce virtually no heat. The light they generate is called cold light, precisely because it is produced without the thermal waste that accompanies nearly every human lighting technology. If engineers could replicate the efficiency of bioluminescent chemistry in artificial lighting systems, the implications for energy consumption, for carbon emissions, for the fundamental economics of illumination would be transformative. The deep ocean has been producing nearly perfect light for an immensity of time. Human technology is still learning how to make a bulb that does not waste most of its energy as heat. Some researchers are pursuing exactly this path. Bioluminescent trees have been proposed as alternatives to streetlights. Bioluminescent coatings have been explored as low-energy alternatives to traditional illumination in buildings and infrastructure. The practical challenges remain enormous. The brightness, durability, and controllability of biological light sources are nowhere near commercial viability, but the principle is sound. Nature solved the lighting problem with 90% efficiency. Humanity solved it with 5% efficiency, and has spent the past century and a half slowly improving from there. The gap between the two solutions is not merely a technical curiosity. It is a measure of how much the deep ocean's biochemistry has to teach us, and how little of that teaching we have absorbed. There is a philosophical pause required here. The deep ocean's bioluminescent organisms have solved problems that human engineers are still working on. They have achieved efficiencies that human technology has not matched. They have developed communication systems that operate reliably across distances, in total darkness, under crushing pressure, with no external power source, no maintenance, and no failure rate that evolution has not already corrected. These are not primitive organisms using primitive chemistry. These are highly refined systems, optimized over an immensity of time, producing results that human technology can measure but cannot yet replicate. The word primitive, applied to deep-sea bioluminescent organisms, is not merely inaccurate, it is absurd. Their chemistry is more efficient than ours. Their signaling is more reliable than ours. Their solutions to the problems of communication in hostile environments are more elegant than ours. If any species on this planet deserves to be called technologically sophisticated, it is not the one that burns fossil fuels to produce light at 5% efficiency. It is the one that combines a substrate and an enzyme in a cold reaction that converts 90% of its energy into photons. The deep ocean has been running a more advanced lighting system than anything human civilization has ever produced. It has been running it since before human civilization existed. There is a second philosophical pause. The deep ocean did not develop bioluminescence for our benefit. It did not refine its chemistry to provide humanity with inspiration or raw materials. The fact that GFP transformed molecular biology, the fact that bioluminescent efficiency shames human engineering, the fact that the biological pump helps regulate the planet's climate, these are consequences that matter to us, but they are incidental to the organisms themselves. The jellyfish did not produce GFP so that Shimomura could win a Nobel Prize. The hatchetfish did not evolve counter-illumination so that military engineers could study camouflage principles. The lanternfish did not join the diel vertical migration so that the planet's carbon cycle would remain in balance. These organisms are living their lives, following the imperatives of survival and reproduction, in an environment that predates human awareness of it by an almost incomprehensible margin. We are the ones who assign meaning to their activities. We are the ones who extract utility from their chemistry. We are the ones who worry about the consequences of their decline. They simply glow and live and die and glow again, as they always have, in the darkness that is their home and their medium and their stage. The biological pump, the process by which organic matter produced in the sunlit surface ocean sinks to the deep ocean, carrying carbon with it, is intimately connected to bioluminescent organisms. The diel vertical migration, that nightly river of living biomass rising to the surface to feed and descending to the deep to rest, plays a significant role in the transport of carbon from the atmosphere to the deep ocean. Organisms that feed at the surface metabolize that food as they descend, producing fecal pellets and respired carbon dioxide at depth, effectively pumping atmospheric carbon into the deep ocean where it can remain sequestered for centuries or longer. The total contribution of the diel vertical migration to the global carbon cycle is still being quantified, but current estimates suggest it transports between 1 and 3 billion metric tons of carbon annually from surface waters to the deep ocean. This is a significant fraction of the total biological carbon pump, and it is driven largely by organisms that are bioluminescent. The same organisms that light the deep ocean with their chemistry also help regulate the planet's climate by transporting carbon out of the atmosphere and into the abyss. If deoxygenation, warming or acidification disrupts the diel vertical migration, if the organisms that drive the biological pump are reduced in number, altered in behavior, or shifted in their vertical range by changes in ocean chemistry and temperature, the consequences would extend far beyond the deep ocean. They would affect the global carbon cycle. They would affect the rate at which carbon dioxide accumulates in the atmosphere. They would affect the pace and trajectory of climate change itself. The bioluminescent organisms of the deep ocean are not isolated curiosities. They are participants in planetary scale systems. They are linchpins in cycles that regulate the chemistry of the atmosphere, the temperature of the surface, and the habitability of the planet. What happens to them happens, eventually, to us. The connection between a luminous copepod flashing in the bathypelagic zone and the concentration of carbon dioxide in the atmosphere above a city thousands of kilometers away may seem tenuous, abstract, impossibly remote. But the connection is real, it is physical, it is measurable. The copepod's migration, repeated by billions of other organisms every night across every ocean, moves carbon. That carbon, sequestered at depth, is carbon that is not in the atmosphere, warming the surface, melting ice, raising sea levels. The bioluminescent pump, as some researchers have begun to call it, is a planetary service performed for free by organisms that do not know they are performing it, and that will stop performing it if the conditions that sustain them change beyond their tolerance. The margin between a functioning biological pump and a diminished one is not wide, and the consequences of crossing that margin would be felt not in the deep ocean, where few humans would notice, but on the surface, where all humans live. And yet funding for deep-sea bioluminescence research remains a fraction of what is spent on other fields of biology. The total annual investment in deep-sea exploration across all nations, all agencies, and all institutions is less than the cost of a single space shuttle mission. The instruments that could observe the deep ocean continuously The cabled observatories and autonomous vehicles and ultra-sensitive cameras that could fill in the blank spaces on the map of deep-sea life exist as prototypes, as pilot projects, as proof-of-concept demonstrations, rather than as the permanent, comprehensive monitoring networks that the scale of the problem demands. We are, as a species, aware that the deep ocean matters. We are aware that it contains life we have not documented, chemistry we have not characterized, processes we have not understood. We are aware that it is changing in response to our actions. And we are choosing, through the allocation of our resources and the direction of our attention, to look away. Not deliberately, not maliciously, simply because the deep ocean is hard to reach, expensive to study, and easy to forget. But the light persists. Beneath the surface, beneath the reach of sunlight, beneath the awareness of most of the humans who walk the continents above, the deep ocean continues to glow. Its organisms continue to flash, to pulse, to signal, to communicate in a language of photons that has outlasted every civilization that ever flourished on the land above. The light does not wait for funding. It does not pause for policy decisions. It does not dim because the instruments are not yet deployed. It burns, cold and constant, in the darkness, as it has always burned, as it will continue to burn, long after the questions we have asked about it have been answered or abandoned or forgotten entirely. There is a world beneath the world we know. It is not a world of darkness, though no sunlight reaches it. It is not a world of silence, though no sound from the surface penetrates its depths. It is not a world of emptiness, though every textbook ever written about it has begun with the word barren or lifeless or desolate. It is a world of light, self-made light, light that is born from chemistry, carried by living tissue, shaped by necessity, and sustained by an engine of biological innovation that has been running, without interruption, for longer than the mountains on the surface have existed. 3,000 feet below the surface of the ocean, the last photon of sunlight has been absorbed. The temperature hovers a few degrees above freezing. The pressure would collapse a human lung in an instant. There is no weather, there is no wind, there is no horizon. There is only water, extending in every direction, above and below, forward and behind. And in that water, in that darkness, in that silence that is not really silence at all, something is glowing. Something has always been glowing. Something will continue to glow long after the last light on the surface has been extinguished. The organisms of the deep ocean have taken the one thing that the abyss denies them, light, and they have learned to make it themselves. They have not merely survived in the darkness. They have flourished. They have diversified. They have built a communication network more extensive than any that human technology has ever created, spanning every ocean basin, involving trillions of individual participants, operating continuously, day and night, for an unbroken span of time that renders all human history a footnote. The language they speak is photons. The medium through which they speak is water. The message they carry is survival. And the conversation, the vast, slow, luminous conversation of the deep ocean, continues without pause, without interruption, without any need for human understanding or human approval. We came to the ocean's edge long ago. We built boats, we cast nets, we mapped the coastlines and charted the currents, and named the surface creatures, and told ourselves that we knew the sea. But we did not know the sea. We knew only its face. We knew only the thin, sunlit membrane that separates the atmosphere from the abyss. Beneath that membrane, the real ocean begins. The dark ocean. The deep ocean. The ocean that contains most of the water, most of the volume, most of the life, and most of the mystery on this planet. And we have barely begun to understand it. The bioluminescent world beneath 3,000 feet is a testament to something that human beings struggle to accept. The universe does not revolve around us. The planet does not belong to us. The majority of life on Earth has never seen us, never been affected by us, never acknowledged our existence. It lives in a world that we cannot reach without extraordinary technology, a world that we cannot survive in without extraordinary protection, a world that we cannot observe without extraordinary effort, and it does not care. The deep ocean is not waiting to be discovered. It is not hiding its secrets, hoping that one day a human submersible will descend to the proper depth and ask the right question. It is simply being alive, being luminous, being itself in the darkness as it has been for longer than we can comprehend. There is something humbling in this. There is something necessary in this humility. In an age when human beings have reshaped the surface of the planet, altered the chemistry of the atmosphere, and measured the distances between galaxies, the deep ocean remains a reminder that the world is not fully known, that the map is not the territory, that there are places on this planet, vast places, inhabited places, luminous places, that exist beyond the reach of human knowledge and human control. The deep ocean does not submit to our instruments. It does not cooperate with our timelines. It does not reveal itself on our schedule. It reveals what it chooses, when it chooses, to whoever is patient enough to sit in the darkness and wait. And what it reveals is light, always light. Light in the jaws of predators, light on the bellies of fish, light in the trailing arms of siphonophores that stretch for 40 meters through the blackness, light in the mucus of vampire squid, light in the spewing defense of shrimp, light in the lures of anglerfish, and the photophores of lanternfish, and the comb rows of catenophores, and the parapodia of tomopterid worms, light in every conceivable form, in every conceivable colour, for every conceivable purpose, and for purposes that are not yet conceived. The deep ocean is not dark. The deep ocean has never been dark. It has been lit, from within, by the organisms that call it home, in a display of biological creativity so vast and so varied that the sunlit world above seems monochrome by comparison. We have looked upward for wonder. We have aimed our telescopes at the stars and our probes at distant planets, searching for light in the cosmos, searching for signs that we are not alone in the universe. And all the while, beneath the waves, beneath the reach of our attention, beneath the threshold of our awareness, something has been glowing, something has been communicating, something has been conducting in light the oldest and most extensive conversation on Earth. We have been searching the sky for signals while standing on an ocean full of them. The deep ocean asks nothing of us. It makes no demands. It issues no invitations. It is not a frontier waiting to be conquered. It is not a resource waiting to be extracted. It is not a puzzle waiting to be completed. It is a living world, older than ours, larger than ours, more complex than ours, conducting its affairs in light and darkness, with a quiet competence that needs no human witness. But we have witnessed it, in fragments, in glimpses, in the trembling footage of ROV cameras and the patient recordings of seafloor observatories and the astonished notes of scientists who descended in steel spheres and saw through tiny windows a world they could not have imagined. And having witnessed it, we cannot unwitness it. The knowledge that the deep ocean glows, that it communicates, that it sustains a web of biological light more extensive and more ancient than anything on the surface, that knowledge changes something. It changes how we see the planet. It changes how we see ourselves. It changes the weight of the word darkness and the meaning of the word light. The light in the deep ocean does not flicker with uncertainty. It does not hesitate. It does not wonder whether it will be seen or understood or appreciated. It burns because burning is what it does. It signals because signaling is what it was made for. It illuminates because illumination, in the deepest and most literal sense of the word, is the purpose for which the chemistry of bioluminescence was refined across an unthinkable expanse of time. The light does not need us. It has never needed us. But we need it, even if we do not yet know how much. Because the deep ocean's light is more than a biological phenomenon. It is a mirror. It reflects back to us the limits of our knowledge, the narrowness of our perspective, the smallness of our experience relative to the vastness of the world we inhabit. It tells us that the planet we thought we knew is a planet we have barely begun to explore. It tells us that the life we thought we had catalogued is a fraction of the life that exists. It tells us that the darkness we feared is not empty but full, full of life, full of light, full of conversations we cannot hear, conducted in a language we cannot read, by organisms we have not yet named. The deep ocean glows. It has always glowed. It will continue to glow when every question we have asked about it has been answered or forgotten. And the glow is not a mystery to be solved. It is a fact to be witnessed, a reality to be respected, a reminder, persistent and luminous and unignorable, that the world is deeper than we think, darker than we imagine, and more alive than we have ever dared to believe. The deep ocean will not reveal itself all at once. It will not give up its secrets in a single expedition or a single lifetime or a single century of research. It will continue to glow in the dark, in the pressure, in the silence, offering its light one photon at a time to whoever is willing to descend, to wait, to watch, and to accept that what they see is only the beginning. Beneath the surface, in the cold and the pressure and the silence, the light goes on.